the assembly of subunit proteins of photosystem 1. Okay, how these complicated complex are synthesized, and how many components are assembled into a functional structure. So it was very difficult to analyze the molecular mechanism of complex assembly, because this process occurs very rapidly. So you cannot follow, okay? It's difficult to follow the assembly process. It's very short time. So we focused on the factor, assembly factor, to find out assembly factor that assist assembly process. Then how this assembly factor works, if we know, we can follow the assembly process. And because this PS1 complex has many subunits, so it was we could expect that there are many factors should assist this assembly. Not only one, more than one, probably many. So today I'm going to talk of several factors we found, but these are not everything. There are many still remain to be found. Okay, so I talked yesterday that this assembly factor had not been found before because probably the strategy to identify the assembly factor is you isolate, for example, the mutant deficient in PS1. But in many cases, most cases, the deficient in PS1 mutant is usually translation mutant, okay, or transcription mutant, or some mutant lacking one of the important component of PS1. So it was difficult to isolate the mutant deficient in assembly. But as I told you before yesterday, by chance when we were working on hypothetical chloroplast openly different YCF3 and 4, we knocked out this by chloroplast transformation to know what these two openly different are doing. Then by chance we found that the mutant of YCF3 and YCF4 or YCF4 deficient mutant completely lacked the photosystem 1. And these two components are not involved in expression of PS1 component. I mean messenger RNA normally transcribed messenger RNA coding for PS1 component were normally expressed, and translation was not affected. So these two factors are not involved in transcription or translation. Probably most after post-translational events. That means most likely the assembly process. Okay, so I didn't show you this data yesterday. When you look at the immunoblotting data using PS1 reaction center protein antibody PSA-A, wild type is 100%, 10% is the dilution. Okay, and then YCF3 mutant or YCF4 mutant didn't accumulate PS1 protein at all. No, even the 10% level is still we could detect the signal, but it's much less than 10%. So it's almost nothing accumulated. No PS1 accumulated. Okay. So then yesterday I talked But we don't know how this YCF3 or YCF4 act in the assembly or post-translational event of PS1. How? We don't know. So what we did was affinity purification of YCF3. So this was strategy of affinity. We just transformed the YCF3 genes with HA tag. So no wild type YCF3 was present, but this HA tagged YCF3 is present in this mutant. Then we did we purified solubilized the cytochrome membrane and then purified YCF3, and then I showed you before these two proteins were purified: YCF3, HA tagged, and interacting protein YCIP1. And also very small amount of PS1, but you couldn't detect by staining the polypeptide. But the immunoblotting showed that the PS1 is a little bit present, the PSAF, but not as a peripheral. Then we did pulse chase labeling. Then after pulse labeling, YC3HA preparation contained pulse labeled PS1 reaction center and also PSAF a little bit. But during a chase experiment, this label disappeared. Okay, so that means that the binding, a small amount of PS1 associated with YCF3 is transient. Okay, the steady state level is small amount of the steady state level of PS1 is present, but this small amount of PS1 is turning over very rapidly. Okay, that is a. So only newly synthesized PS1 associate with YCF3, or YCF3 associate only newly synthesized PS1. That's why this protein YCF3 act in a post-translational event of PS1 synthesis. Okay, this is the way. The same thing, we did the same strategy to analyze other factors. Okay, this is important. If you understand this, you can easily understand the following experiment. Okay, so just look at the this polypeptide pattern. No, very simple, right? Many minor amount of PS1 and part of reaction center PS1 and PSAF, but not other PS1. So this is Y3IP1, so I skipped this. I talked yesterday already. So our conclusion is YCF3 is interacting with the initial assembly steps of reaction center of PS1. Okay, then affinity purification of the YCF4. No, another hypothetical group. Openly different. Of course, this knockout this gene result in no accumulation of PS1 like YCF3. So we purified, using after tagging YCF4 with HA, and then purified. Of course, after we isolated thylakoid and the solubilized thylakoid and affinity purified. This is a little bit small. This is better. Okay. So this is a result of purification. Okay. It's a little small. This is A3 is PS1 purified PS1 PS1 complex. So to show the position of PS1 and LHC1 polypeptide on the gel. This is Y type is a negative control, so we used Y type at almost no protein was recovered, indicating this positive HAYC4 is the polypeptide recovered is very specific. Okay. So if you look at this polypeptide, this is a PS1 reaction center here, diffuse band. There are many minor contaminations somehow, but it's some PS1 related proteins you can see. You see? PSA C, E, PSA L, PSA DF, and also some LSC1 proteins also recovered, which means that YCF4 interact not only PSA A, B, PSA F, but additional PS1 polypeptide and LSC1 as well. These are confirmed by immunoblotting PSA A, C, D, F, L, no PS2, almost nothing, and some LSC1. So my question to you is, what this difference of the polypeptide pattern compared with purified YCF3 polypeptide pattern means? No? Why YCF4 associate more PS1 protein, even the LSC1, compared with purified YCF3 preparation? What this means? What you can extract, conclude from this data for comparison with the data between the YCF4 and YCF3? What you can speculate?
YCF4 is critical for PS1.
Yeah, critical, of course, critical. YCF3 also critical. YCF4 is critical. No, if you knock out, no PS1. But the difference, we observe the difference between YCF3 preparation and YCF4 preparation. This difference, what this difference means, or suggest, or tell us what. Okay, is there anyone?
Major role in capturing light harvesting.
Light harvesting?
YCF4 plays major role in harvesting capturing light.
Now, LH1
Yes,
is
yes.
also present. Why? It's okay. Okay, just think. Okay, so then speculate by yourself, and then I explain. Okay. This also, I did a immunoplotting, pulse chase labeling as we did for YCF3. Okay, the same pulse and chase. Okay, and PSAA very strongly labeled in this time, and also PSA C, L, DF. This is YCF4, LSCA1, LSC1, something. So and also doing a chase, PS1, no, disappeared, going down the level. And PSA DF, it's also going down, okay, decreasing. So these PS1 proteins associated with YCF4 are also transient. Okay, transient. It's not stably associated with YCF4. The newly synthesized PS1 proteins are preferentially associated with YCF4, like YCF3 also. So that's why we can conclude that the YCF4 is also involved in post-translational event of PS1 accumulation. Okay? And also, this preparation was slightly green. Okay. YC3 was not green. Preparation was not green because probably there was a PS1, but its amount was so small. But this time, no, the protein level of PS1 is more abundant. So then we can measure the absorption spectra of this YC4 preparation. Then, no, CoQ-A, this one, and this part is carotenoid. Can you see it's small? And also the peak of the red region is 678 nanometer. This is the typical absorption peak of photosystem 1. Photosystem 2 is 670 nanometer. So this is not PS2. Or LSC1 or 2 also a little bit shorter. So this is the typical PS1. The chlorophyll associated present in this preparation is derived from PS1 complex. And the HPLC, high performance liquid chromatography, indicated that chlorophyll A is here. A little bit beta carotene and slightly chlorophyll B. Okay, because it contains a small amount of LSG1, so PS1 core lacks chlorophyll B, but LSG1 contains both chlorophyll A and B. So this represents chlorophyll small, chlorophyll B amount, chlorophyll A, and beta carotene. Beta carotene is present in PS1 core and LSG1. LSG2 lacks chlorophyll beta carotene, but LSG1 contains beta carotene. What this means? Why this YCF4 preparation contain chlorophylls? What this means? YCF4 associate not only PS1 proteins, apoprotein, but also cofactor are present here. Okay. This is the same. Right. And then we separated this YCF4 preparation on sucrose density gradient to see if the chlorophyll present in YCF4 preparation is really associated with proteins or free chlorophyll or not. Of course, the peak of the chlorophyll absorption peak at 678 nanometer indicate these chlorophylls are not free, not bound to PS1. But to confirm, we purified, separated. This is thylakoid membrane, okay? The thylakoid extract, solubilized with detergent, and LHC2 band and the PS2 PS1 LHC1 band is separated. And HA tagged YCF4 preparation, we see here and here the green band. So indeed, this shows that the chlorophylls recovered with YCF4 preparation is large, in large complex. And the position from the gradient is very close to the PS1 LHC1 supercomplex, and this is PS1 core complex. Because there's no PS2, so PS2 core is separated here, and the PS1 core is separated here, but this contains only PS1. And then we analyze the polypeptide of this band. The Y3 band is colorless, and also immunoblocking. To analyze more in detail the composition of polypeptide. So it's a little bit difficult. Small, so it's complicated. This is a reference of PS1 LHC1. So PS1 LHC1 polypeptide pattern here, and Y1 here. Polypeptide PS1 reaction center, This is YCF4, HA target YCF4, PSA-D, L, E, C. Okay? And Y2 green band, that is heavier and larger size, PS1 reaction center, D, L, E, C, small PS1 polypeptide, and LC1 was there, associated with PS1 core. That's why Y2 is larger than Y1, because the presence of LC1 association. And Y3 is colorless and contain only YCF4. The only YCF4, but still so large, so that means the YCF4 forms the oligomeric forms, large oligomeric oligomer. Okay, is that clear? And the immunoblotting also show that Y1, this one, contain PSA-A, C, D, E, F, but not G, I, not K, L, no LSC1. But Y2 contain PS1, PSA-A, C, D, E, F, I, L, and LSC1, LSCA 1, 2, 3, 4, 5, 6, 7, 8, 9. So LH1, almost all LH1, but the LSCA 2 is very low. 9 is also very low. So LH1, at least 7 different LH1 are present. Okay, now you understand what's the difference between YC4 and YC3 preparation. So YC3 is involved in initial assembly of PS1, particularly assembly of reaction center protein PSA A and B. Okay, okay, and then YC4. This is YC4. YC4 is required for integrating other peripheral small PS1 subunit to form a PS1 core complex, subcomplex. We call it subcomplex. And also, YC4 is involved in the associating of LHC1, LHCA1, 3 to 8, that are located in a 2 layer of the tetramer on PSA F and F side. I showed the day before, yesterday probably, the structure of PS1 and the LSC1 tetramer. For LSC1 tetramer, there are 2 layers on one side. Okay, but there was one dimer, LSCA 2 and 9 were present here, this side. Do you remember that? So in the YCF4 preparation, lacks LSCA 2 and 9. No, only little, very little. And also here, PSA-K and PSA-G are present in the edge of the 2 layers of tetramer. That stabilizes the, but these 2 are still absent. So PS1, LSC1, subcomplex. It's not completed. Okay. So our conclusion is YCF3 is involved in initial assembly. The YCF4 is required from initial to the almost the final assembly process. All the way from beginning to the end. And then finally, dimer of LSC1, LSCA2 and 9 associate this position, and PSA-K and G are present here. That's required for stabilizing LHC, no, 2 tetramers of LHC1, no, this side, is integrated. Okay, so these 2 factors are very essential for this process. And from our data, we could make this working hypothesis. Still my hypothesis, probably. But our data is consistent with this model. But it's not easy to detect this assembly intermediates biochemically, because this process is very rapid. And if you try to look at this assembly intermediate using a wild type chloroplast, bunch of mature PS1, among the huge amount of PS1, mature PS1, only small tiny amount of assembly intermediates are present. So this situation is difficult, right? For example, if this is 1%, so 99% is mature PS1, and only 1% you have to find. It's difficult in a biochemical method. Okay, so this analyzing through this assembly factor was so far rather successful, and I think we need to find out other new assembly factor to completely understand this assembly process. So this the data was this experiment was done by Sridhar Nilpari from here. He was a postdoc in my group some years ago. He finished his PhD here in Hyderabad, and then stayed several years in Okayama. And he did this very excellent work. And another interesting factor was CGL71. This is a chloroplast green lineage 71, number 71. This was also a nuclear encoded factor, also PGY, pale yellow green mutant in vascular plants, Arabidopsis. And also this is also present in, this gene is present in the shell bacteria. So it's conserved in a photosynthetic organism, so it must be very important. So this factor was first reported in shell bacteria, mutant, and also it shows that slightly PS1, the amount of PS1 decreased. But in Arabidopsis, significantly decreased. So this factor may be involved in PS1 accumulation, but we didn't know how this works. So before we starting Arthur Grossman in Carnegie Institute in San Francisco, California, analyzed this CGL71 mutant. And they concluded that this may be involved in PS1 assembly or post-translational level. And also, interesting phenotype is white type. If you grow the cells on agar plate, white type like this, and CGL71 mutant in oxic condition, in the presence of oxygen. This is a photoautotrophic condition. The cell didn't grow. But anoxic condition, it grows photosynthetically. So somehow this mutant is sensitive to the presence of the oxygen. And then PS1 works well in the absence of oxygen. So PS1 must be very sensitive. So what we speculate is what the oxygen affect in PS1 is iron sulfur cluster. PS1 contain 3 iron sulfur cluster or iron sulfur centers that works as an acceptor of electron acceptor of PS1. Stabilize the charge separation and donate this electron to the ferredoxin and finally to NADP plus to produce NADPH that is used for reductants for carbon dioxide fixation. So from this phenotype, we can speculate, somehow acceptor side is damaged in this mutant in the presence of oxygen, but it's stable in the absence of oxygen. So first we analyze the polypeptide accumulation if this presence of oxygen is affect specifically to PS1. So this we tested only PS1 here. A wild type dilution 10, 150, 30, and CDL71 grown in oxic condition. So PS1, PSA-A, C, D, L, subunit are almost missing in the cells of this mutant cell in the presence, grown in the presence of oxygen, but hypoxic. So it's low, very low CO2, O2 condition, nearly anaerobic condition. PSA-A, C, D, L are accumulated as expected from the growth in anoxic condition. Okay. So PS1 is proteins are unstable and in the presence of oxygen and stable in the absence of oxygen. Okay, this was confirmed. So we did the same strategy to tag CGL71 with HA and to purify to see which polypeptide are interacting with CGL71. Okay, the same things. This is purified. There is no negative control data, but negative control data is the same, right, as YCF4, YCF3, no Y type. So we know that there's no almost very low non-specific no recovery of polypeptide. So we found only CGL71 like this. We didn't see any very major proteins interacting. But we tested the immunoblotting data to detect very high sensitivity of the small amount of PS1 protein, whether the PS1 protein is interacting with CGL71. The Y type, almost nothing. And the CGL71HA, we could detect slight amount of YCF4, YCF3, Y3IP1, and PS1 reaction center, but not other small PS1 subunits, PSA-C and PSA-D and E. Why we focus on this is PSA-C associate two iron sulfur centers, FA and FB, as a terminal electron acceptor. So we just suspected that this might be interacting with CGL71, but was not the case. And also, PSA-D and E are also on the peripheral stromal side. PSA-C was covered with these two subunits. So, but these were not interacting. But only reaction center we found interacting. And reaction center contain one iron sulfur cluster, Fx, that accepts electron before Fa and Fb. And just present is between the two reaction center PSA A and B. So maybe this protein is probably involved in stabilization or protection of Fx rather than Fb, Fx, to protect Fx from oxygen. And also, this is power state leveling, and it's not so clear, CDL71 here. And reaction center proteins, this diffuse band is not so strong signal, but it's really newly synthesized reaction center protein worth interacting. Okay? And this is also interesting. CDL71 is turning over during a chase. This protein is turning over, like YCF3 also turning over. YCF4 was not turning over rapidly. So these two are very interestingly, very rapidly turning over. Okay, so this is also three published. He's mainly involved in this work with the collaboration of Arthur Grossman's group. So from this data, we speculate that when the two reaction center PSA A and B proteins assemble in into the cytochrome membrane, as I mentioned, YCF3 and Y3IP1, two factors were involved, and also CDL71 is required. So probably before the on the stromal side, PSA C, D, E assembled. Before here, Fx is assembled on stromal side of this region. But before the PSA C, D, E assembled onto the reaction center, this Fx is exposed, could be exposed to the oxygen. So CDL71 bound to this region. This is a model to prevent the access of oxygen to this iron sulfur cluster, to protect. No. And then PSA D, E, C here is assembled. Of course, in that case, CDL71 is removed, exchanged. Then, once this assembled, Fx becomes stable because oxygen will not access to this site. Okay? So CDL71 is something like this, where it's a kind of dimer, it's a model. And oxygen is there. So iron cluster center is assembled, then this bind here. This contains 1, 2 transfer helices. So it's covered like this. No? That's the model, working model. So
But CDL71 is transit or still it's permanently incorporated?
no, it's once it's this PSA C, D, E is associated, it's replaced by. This also covers this side, right? So the working hypothesis is the same things, but it's CDL71 also works this region, these steps. No, Arabidopsis is PYZ7. Chlamydomonas is CDL71, and Cyanobacteria is YCF37. It's a different name depending on the different organisms, but because of this, it reflects the mutant phenotype, pale yellow green, without PS1, and Arabidopsis doesn't grow without photosynthesis, so it becomes, the seedling becomes pale yellow-green. Shallow bacteria still can grow without this somehow. PS1 level is reduced. It's always the case. Some phenotype is weaker in cyanobacteria and stronger in vascular plant. Okay, so this is a almost. So this is what we have for the moment, and also there are not so many other factors are found. So this I talked about 4 different factors. 2 factors are associated with each other. So somehow major part is, I think, these 4 factors I talked is very major assembly factors. You can see CD71 contains small amount of YCF4, 3 YCP1, so it's separated mostly, but YCF3, YCF4, YCP1, CD71 are mostly closely associated. But if you purify, the interaction is not so strong, so it's separated. But small amount is present. No, negative control has nothing. Okay, so this is not. We don't think it's contamination, but it has some meaning. Okay, so we have no data, but another interesting or important question to be answered for PS1 assembly is how cofactors, see, chlorophylls, carotenoids, and also as a acceptor, there's a naphthoquinone are also present. These cofactors are how cofactors are integrated into PS1 complexes. This is totally remains unknown. This is also very difficult questions. And we try to analyze this process, but so far we didn't have any fruitful results. But in the next lecture today, I will talk about a little bit about coupling between the chlorophyll synthesis and also subunit assembly. Okay, so I'll stop here, and also very much welcome your questions or small c. Here? C071? HA? Which one? And? Where? Oh, of course, this is a nuclear encoded, so C071 HA is fused to a C-terminal side of CD71. This is nuclear encoded. But in the case of YCF3 and 4, sometimes we made both fused either C-terminal side or N-terminal side. We made at the same time. And sometimes if you fuse to C-terminal side, the protein may become unstable, or N-terminal side may make a protein unstable. So we did both at the same time to save time, to see which is better, or which is better for purification, or which is better for the stabilization. But in the case of the nuclear encoded protein, we always fuse at C-terminal side. Okay? This is just a strategy. It's not very important meaning, but sometimes if C-terminal fusion at C-terminal side didn't work, so we have to find out a good position in the middle of protein. But that is not easy, right? So that's the case, okay? Because these data are so tiny, detailed, so it's maybe sometimes difficult to follow. But it's if you have a which data, well, it's difficult to understand. If you say me, that helps me to explain better. But okay.
What is that LHC4?
LHC2? Which?
Previous table.
Hmm? Here.
Yes, yes.
Okay, this is thylakoid membrane. So Chlamydomonas has very several LHC2 genes, but some are very, very similar each other. So we categorize major LHC2 as LHC2 type 1, type 2, type 3, type 4. Okay, so this is type 3 and 4. Yeah, it didn't separate, but this position LHC2 type 2, 3, and 4. This is LHC2 type 1 here. It's hard to say type 2. No, type 1. Type I think type 1. It's separated somehow. I don't know. Probably one of this one is type 2 and this one is type 1. Sorry, it's my mislabeling, but anyway, these are all different types of slightly very similar each other, but it's a different type of LHC2 apoprotein. Okay, so this is thylakoid. So you can see from this part. Okay, so I have a question to you. After pulse and trace labeling, the pulse labeling, if you look at this data, could you say something interesting to you? No? It's important to see, if you see the data, wow, this part of the data is interesting, or this part of data is strange. This impression, right? If you look at the data, raw data, this is important for if you do a research. You get some data. Okay, I got the data. That's all. Go to have a coffee. Is that right? No, if you see the data, try to find which part is interesting, which part is unexpected, which is new. It's better to spend some time to speculate. So this is not your data, but if you look at this, you can try to find something you feel unusual or strange or interesting. Is there anything like that? Try to find something you impress you. Anything. Do you have something? After purification, she said, strong signal was found in the label for signal, but we couldn't detect any signal in thylakoid membrane. Why? Okay. Oh, that's a good point. Is there anything You think strange or unexpected or want to know more further? Do you have something? Yeah? Tell anything. No? So, many protein? Some proteins here, which you are impressed? Strong? Which one? Second? D1? Why D1 is very strongly labeled? That is a good point. Something like that. You should know. Just see. There are many bands. That's okay. Complicated. I don't like. But one step further, you should go. Then some bands are diffuse, but some bands are stronger. Some bands are weaker. Something like that. That you should notice to find the new things. Otherwise, you miss the data. And then you bring to your supervisor. I get this data. I don't know this is interesting or not interesting, successful and unsuccessful. I don't know, but supervisor asks supervisor, what do you think? This is not good. It's not interesting or it's not fun. After doing an experiment, if you find something interesting by yourself, that becomes science. Okay? It's not, if you just do experiment, that's a technician's work. Okay? But if you find something from the data, or if you try to explain something from the data, that becomes a scientist. Okay? So the question first, why? After purification, strong signal detected, CD71, but not in the thyroid membrane. This is very important. This affinity purification so excellent. You can concentrate significantly, so signal becomes so strong. But here, the amount of CDR71, catalytic amount, very low. So this should be signal, but you couldn't detect because of the sensitivity. But if you detect too strongly, so other protein also appearing. So this signal may be covered by other signals. So that's why it is strong signal, but okay. So another interesting suggestion is why D1 is strongly labeled. There are two possibilities. One is this is not labeled with carbon 14, but it's sulfur 30, S35. So if the protein lacks methionine and cysteine, you can't label. Okay, and also if the protein has a bunch of cysteine and methionine, it's strongly labeled. But this is not the case. D1 is another possibility is protein is abundant and synthesized very rapidly. Okay, so then you will get strong signal. Or the protein is not abundant, but it's turning over very rapidly. So it gives you a strong signal. In the case of D1, the amount of the protein is much, much lower than LHC proteins, but still strongly labeled. And D1 has not so many cysteine or not so many methionine. So this is, as you know, D1 is turning over very rapidly under illumination. So if you don't illuminate the cell during a pulse labeling, D1 is normally labeled very like other proteins. So this is strong. And LHC is very abundant, most abundant proteins, but labeled strongly relatively, but it's still compared to the abundance of LHC2 protein in the thyroid membrane relatively. So the synthesis of LHC2 is not very high. And also these are two bands here. So this is ATP synthase alpha beta terminate. This is also somehow always strongly labeled. That is the case. Okay, PSA B, not really. You don't see in the thyroid membrane. It's not, sensitivity is not high compared to PS2. PS1 and PS2 accumulation level is nearly the same, but PS2 is more strongly labeled. So PS2 turnover, PS1 doesn't turn over. Okay, that is how to see the data. So now if you do your own experiments, so after getting the data, you should take time to see carefully data, and then think about from which what you can extract. This process is very important. Okay. Any other questions? No? If not, I stop my talk.
Thank you, sir, for the wonderful talk.
Yeah.
Anoxic conditions.
I need to ask three, but it's probably it's difficult to do anoxic condition for pulse labeling. Pulse labeling is start from very small. Can be. And if you add the glucose oxidase system in the liquid, so glucose oxidase oxidize the glucose consuming oxygen. So the concentration of oxygen in the medium will be reduced. Good point. I have to check manuscript, but it's very, very important service. Here, like this. Okay, thank you. Do you hear me well? And in this talk, I going to focus on the chlorophyll synthesis. Okay. And as I talked to you before, the greening of plants, the vascular plants in general, if you grow in the dark, it becomes etiolated. And then if you grow them in the light, it becomes green, fully green. So here, etioplast, cell contain etioplast, and here it's chloroplast. So I introduced chlamydomonas greening, and the chlamydomonas is, as I told you, that accumulate chlorophylls when grown in even in the dark, okay? Because it contains light-independent protochlorophyll reductase. So it can synthesize, the cell chlamydomonas can synthesize chlorophylls in the dark. But if you illuminate the cell, of course, synthesis rate of chlorophyll enhanced. So the cell becomes more green. But you can grow the green cells in the dark. But I talked before, the Y1 mutant is yellow in the dark mutant, like a vascular plant. And this mutant cannot synthesize chlorophyll in the dark because light-independent protochlorophyll oxidoreductase was deficient. It's deficient. So only light-dependent oxidoreductase is present. So if you grow the cells in the dark, it becomes a yellow cell, and the chloroplast become etioplast. Okay, and this advantage of chlamydomonas is if the dark grown cell is shifted under the condition of light, it starts to synthesize chlorophylls, and then about 8 hours, it becomes fully green. It's okay for me. Okay. Okay, so so we can follow the process during a greening using this Y1 mutant of chlamydomonas. Okay, so they are showed before, but just remind you of the process. So from zero, dark green, dark grown Y1 cells, yellow cells, and then greening, you extract the pigments, and then measure the absorption spectra. Then 0 hours, there's no chlorophyll. Okay. Okay. This is convenient. Like this. You can see better. No chlorophyll accumulation of carotenoid. And during illumination, the chlorophyll accumulate. Okay? Like this. Okay. And chlorophyll binding protein like photosystem 1 and photosystem 2 and LSG, doing a greening, yellow cells, you don't see these chlorophyll binding proteins. But during a greening process, PS1 accumulate, PSAA, these are all PS. LSG1, PS2, LHC2 accumulate. Although I told you that the cytochrome B6F complex contains one chlorophyll molecule, but this is PET A is cytochrome F. Although the amount is lower, but still accumulating even in the yellow cell. That is an interesting point. We have not characterized further, but in the absence of chlorophyll, B6F is still at a reduced level, but it accumulate. They increase it like this. Okay, that's interesting point. So, how the pigment accumulation occur? So, this is just absorption. The mixture of chlorophylls and the carotenoids. We don't know which component accumulating. So we analyzed pigments by HPLC. We extract the pigments with organic solvent and analyzed composition of pigments by HPLC. This is the elution time and also absorption 270 nanometer, 430 nanometer, 143 nanometer, depending on the component absorption peak are different. So you can choose the detection wavelengths. This is an example. For example, 30 is you can detect the chlorophylls and the carotenoids. So this is the elution time. So first, hydrophilic xanthophyls, rhodozanthine. This is a craminomerase. So rhodozanthine, neoxanthine, violozanthine, lutein, and chlorophyll B, chlorophyll A, alpha, beta carotene are separated like this. Okay. And if you want to analyze the quinone, 247 nanometer, you can detect a very small peak of plasquinone here. And naphthoquinone, you can detect here. In the craminomerase, hydroxyphyroquinone is present in PS1. This is indicate the measure of PS1 content because this quinone present in photosystem 1. And plasquinone is present in cytochrome membrane, bilayer, lipid bilayer, and photosystem 2, both. And during illumination, we measure this component, okay? The chlorophyll A and B, you can detect here chlorophyll B and the chlorophyll A. It's a huge peak. It's easy to detect. So during illumination, chlorophyll A increases like this, and chlorophyll B increases like this, up and down. And hydroxyphyroquinone, this is associated to PS photosystem 1. It's increasing. Oh. Oh. Like this. And plasquinone going up like this. And this is the carotenoid. Alpha carotene, beta carotene is a carotene without oxygen atom, so carotenes. And lutein, rhodozanthine, violozanthine, neoxanthine is the xanthophyls containing oxygen atoms. So this is a little bit more hydrophilic compared to this carotene. That's why the alpha and beta carotene eluded later than xanthophyls. So this is the changes in this carotene. Okay. Rhodozanthine is somehow after lag phase, it increases, but the beta carotene decreases, and violozanthine increases, and alpha carotene almost constant. Rhodozanthine, neoxanthine, xanthophyls are increasing. Okay, these are mostly associated with light harvesting complex. So probably this reflects to accumulation of light harvesting complexes. However, although here chlorophyll A and B are absent, almost absent, but if we look at carefully, this is after 5 hours greening cells, we detected many chlorophyll-like peak by HPLC profile. You see here. Chlorophyll A is here, chlorophyll B is here, okay? And chlorophyll A type peak, A1, A2, A3 were also separated before the chlorophyll A separation, okay? And chlorophyll B, in addition to chlorophyll B, chlorophyll B1, B2, B3 were separated somehow. And if you look at the absorption spectra of this, chlorophyll A, A1, A2, like that, the absorption spectra was exactly the same. Okay, chlorophyll B type also the same. There's no indication that A1, B1, B2, B1, B2, probably this is B3. Okay, the same. But in the separated in a different position. That's happened so well. So protochlorophyll A is converted to chlorophyllite, and then converted to chlorophyll A and chlorophyll B. But there are additional steps. This is called chlorophyll A GG, chlorophyll A DHGG, chlorophyll A THGG, and then chlorophyll A. So this is an abbreviation of, this is granule chlorophyll A. And dihydrogranule chlorophyll A. Tetrahydrogranule chlorophyll A. And the same series of chlorophyll B type also present. So this is A1, corresponds to A1 here. And then the dihydrogranule chlorophyll A is here. And then tetrahydroxy granule chlorophyll A is here. And then finally, chlorophyll A. So this is a stepwise conversion to. So this usually in the white type cells, this component you could detect small amount, negligible amount. So usually we don't care very much, but doing a greening experiment, so it's starting from here, so it's substantial amount of this intermediate or derivative of chlorophyll A and B accumulate during the greening cell. Okay, this happened. So granule reductase is involved in this conversion. This is also granule chlorophyll is this is a phytol, but granule chlorophyll A has additional this double bond here. So granule reductase stepwise this double bond is hydrolyzation, adding a hydrogen atom to remove this double bond. Then finally, only one double bond remained in a phytol chlorophyll A. So 1, 2, 3 steps, okay, occur. Also, this enzyme also involved in alpha tocopherol synthesis. This is also granule. Okay. Sorry. Also, naptoquinone, menaquinone, vitamin K, pyroquinone, pyroquinone, also the same structure is also not reduced. These are involved. But now we focus on this part. So there's a, in parallel, these granule reductase occur in a granule. This is a precursor of phyto. The granule is here. The granule portion to connect it. So that's why this is granule code. And pyrophosphate. So this is a phosphate. So the granule reductase is CHLP gene or proteins. This is CHLP gene encode this enzyme. And this also granule pyrophosphate is reduced to phytol pyrophosphate. This is phytol. Okay. So this acts in this process, and this is chlorophyll A, and this is bind to chlorophyll A. This phytol may bind to phytol to make chlorophyll A, or granule pyrophosphate react with chlorophyllide A, and then to form chlorophyll A, granule chlorophyll A. And then this is reduced to chlorophyll A. So that two pathways occur. It is proposed that two pathways occurs. Okay, we don't in our this screening experiment we cannot distinguish between these two pathways. So we just make a graphs of this our results during a greening. This happens that the greening yellow cells, and this is fully green cells. So chlorophyll A is accumulated like this. There's a lag phase here, 1 to 2 hours. So in the beginning of 2 hours, almost no chlorophyll A and B accumulate, and then start to increase. But before the chlorophyll A accumulation, granule chlorophyll A starts to increase, and the peaking at 3 hours later of the illumination, and then slowly going down. And then the granule chlorophyll A increases a little later, and then peaking at 5 hours, and then going down. And tetrahydrogranule chlorophyll A also going up, and then peaking at 5 or 6 hours, and then going down. Oh. Similar pattern was observed for chlorophyll B derivatives. Okay, so this happens. So doing a greening, we cannot say very clearly, but it's a greening. This process occurs. Okay, and also chlorophyll B is we don't know exactly. The chlorophyll B is usually synthesized from chlorophyll A, but here even granule chlorophyll A is converted to chlorophyll B, granule chlorophyll B, and this reaction also occur, and also this reaction also occur. So this is happening. This mutant. So just want to check. Okay. So doing a greening in at least this chlamydomonas Y1 mutant, the accumulation pattern of chlorophylls are a little bit different from the wild type, and abnormally this chlorophyll A derivative and chlorophyll B derivatives accumulate transiently. This happens. Okay. Then we analyzed chlorophyll binding proteins. It's gradually accumulated, as shown in the beginning of the Western blotting. But here we show the complex, no, not apoprotein accumulation, but complex accumulation is analyzed. Not every time, because every hour, because it was so hard. So we tested 2 hours and 10 hours after 10 hour, 2 hours, and 10-hour illumination. This is very initial illumination. So it's very low amount of chlorophyll accumulated. And this is free wind. Okay. So here, the most cells are still yellow, so it was not easy to accumulate the cytochrome membrane from these cells. So we have to start from the plant, huge amount of culture. And then we collected the membrane, break the cells and collected the membrane fraction and solubilized and separated by Shuko-Sensi gradient ultracentrifugation, as I showed many times already. So this is a gradient of top of the gradient and 0.1 molar sucrose, and this is the bottom of the gradient and 1.3 molar sucrose. So smaller complex separated here, and the larger complex separated here. So you see the difference. We try to load a similar amount of the chlorophyll, but obviously this is less. But the pattern looks different. It's not the same. See? So this is we fractionated this gradient from the bottom, fraction number, and then we took the absorption of the fraction. This is a 675 nanometer. That means chlorophyll peak, chlorophyll absorption. Okay, so from the 10 hours, it's a typical pattern like wild type. A1, A2, A3. This is LHC2, PS2, or PS1 core. And this is PS1 LHC1 super complex. But in a 2 hours illumination, the complex formation was incomplete. So we could detect A1, A2, a very small amount of A3. See, only. We call this peak, this is shoulder. Okay, after peak, it's the shoulder. And then we analyzed all the fraction. We analyzed the polypeptide from all the fraction. And we detected this in the immunoblotting because the protein was not sufficient. So this is the 10 hours and the 2 hours. Okay, only this part, right? So 10 hours, the A3 fraction was here, PS1, a little bit PS1 in A2, and PS1, LHC1. We didn't detect the PS2 antibody, but this is obvious PS2. And also in 2 hours, already PS1 is accumulated, but the separated mainly A2 fraction. Here, okay, here. Now, after 10 hours, you detect the signal in A3, but here A2. And also here, PSA A, D, G, LHC1 was mostly separated at the same position. But here, small amount of PS1 is present in A3, and PSAG is here. But A2 major PS1 was separated in A2, and the PSAG is here, but PSAG was not here. and LHC5 was a little bit separated very broadly. We couldn't detect only small amount. Signal was so weak we couldn't detect, but probably PSA5 also present here, but signal was so weak. So A2 here, A3 is here. Then we analyzed the pigment in this A3, A2, A1 fraction. Okay, this is not the total. This is a pigment composition in each chlorophyll protein complexes. A1, A2, A3. And also doing illumination, how it changes. So chlorophyll A is as expected, chlorophyll A increases. Doing illumination in A1 fraction, chlorophyll A increases. And granular chlorophyll A, this is the first one, go down. And chlorophyll dihydrogranular chlorophyll A go up and transiently go up and down. Tetrahydrogranular chlorophyll A is also same things. Okay, the pattern is very similar. A2 fraction, this is PS1 core and PS2 core, also the same. And A3 also the same, very similar pattern. So each chlorophyll protein complexes associate in doing intermediate illumination, associate both chlorophyll A and chlorophyll A derivatives. And chlorophyll B also the same, very similar. So this suggests that chlorophyll A, not only chlorophyll A and chlorophyll B, but granular chlorophyll A and B are integrated into chlorophyll protein complexes without waiting. No, the granular chlorophyll A is converted to chlorophyll A because these are very hydrophobic, so it's already integrated. So immediately this synthesized, but it's converted to chlorophyll A in the chlorophyll protein complexes, right? So somehow the granular reductase convert these derivatives present in the chlorophyll protein complexes. So we suspected that some pigments are present inside the protein complexes. So enzyme granular reductase may not access to the these granular chlorophylls, but it's actually it seems that it's access to in the complex. Okay, so that's the. I think I didn't put. Okay. So this is a chlorophyll synthesis, but this is the very last steps of the chlorophyll synthesis. One of the last steps of chlorophyll synthesis is the binding of the phytol or granular chlorophyll to the chlorophyllite. So these steps makes pigments hydrophobic. So this cannot be chlorophyll cannot be present in soluble state. state. So it must associate into chlorohyl protein, chlorohyl binding protein to form complex. So we expected this process may be somehow coupled with chlorohyl protein complex assembly. But so far we haven't clear data that show that it's coupling. So I didn't include in this slide. Maybe next lecture we are going to talk about the mutant of this granule reductase deficient mutant. I didn't prepare today, but the next lecture probably I can talk to. So this is also we wanted to try to analyze the molecular mechanism that couples the subunit assembly and cofactor assembly, the chlorohyl assembly, how it works. So probably this the chlorohyl synthesis the last steps of the chlorohyl synthesis must be coupled with this assembly process. But so far we haven't obtained the positive data showing this coupling for the moment. And also we next lecture I'm going to show this granule reductase deficient phenotype, but still chlorohyl protein complex are assembled. So far it's difficult to analyze. Okay, so this is just greening and detailed greening process. So I just add some additional data again about the assembly of PS1 complexes. I show you this data before, and how do you say? I didn't tell you a detail, so I show you detail now. This is a separation. This is wild type. Okay, the separation pattern of chlorohyl protein complexes that you saw many times already. LHC2, PS2, and PS1 LH1 supercomplex was separated like this. And we separated these chlorohyl protein complexes from the cells growing logarithmic phase. Logarithmic phase means the cell is dividing very actively. That means the cell synthesizes proteins actively. So, for example, PS1 proteins are actively synthesized. Okay, the activity of the assembly is active. Also, this is separated from the cells. That was the growth of each growth is stationary phase. So it doesn't divide anymore, but still cell is alive, but do not synthesize the proteins actively. It's very protein synthesis activity is very low. So PS1 assembly is very low, almost stopped. Okay? And we separate it. Looks very similar. Okay? It's not interesting. But we fractionate it. Okay? And the peak of chlorohylus was addition pattern. It shows A1 and A3 is almost the same, but we see a slight difference in the intensity of the H band. If you look at here, you can see the difference. Here, it maybe had a little bit weaker. You see, this small change was very important. Then we fractionated this. Okay, this is LHC2. You can see 25 to 30 kilodalton protein here. This is type 1, type 3, type 4 LHC2 apoprotein. This is PS2 proteins. And this is PS1 proteins. Okay? Major, minor, small subunit. And LHC1. And we very much carefully analyze the polypeptide pattern by immunoblotting. This is staining, but this is immunoblotting. Okay? So from the logarithmic phase, the cell from the logarithmic phase, you see a PS1 here, A3 band, here, right? Strong band. All the subunits and LHC1. But this is a A2 is PS2 proteins here. Okay? But if you look at carefully, there is a minor amount of PS1 is observed here in A2. Not PSA G and K. Okay? G and K I talked before. So this is the same, the difference. So we thought two possibilities from this data. Okay? One is these are just when we solarize cytokine membrane artificially, some of the subunit was dropped off, and then a smaller complex was generated artificially. So this fraction was because of the detergent. PS1 complex was becomes subcomplexes, smaller complexes. So it doesn't mean anything. But if you look at the separation pattern from the cell from the stationary phase, we didn't detect any minor PS1 here. So this suggests that, this could be present only the cells that the actively synthesize photosynthesis apparatus. So suggesting that this could be assembly intermediate. Usually we don't detect because assembly process proceed very rapidly. Okay. So I showed before this pulse test labeling, and I didn't explain detail. So if this is not artifact, this is a newly synthesized subcomplex in the intermediate subcomplex. After pulse labeling, the protein PS1 here may not be labeled, but here it's labeled more strongly than others. That is a hypothesis before we start the experiment. Okay, so this is the result of pulse labeling. Because for the radioisotope work, we used a very small scale experiment. So this is after pulse labeling and pulse labeling and chase experiment. We separated by Shuku centigrade the chlorophyll protein complex, but using a very small tube. Now we wanted to use a large tube like cold experiment, but it was not possible because we have to work in a separated facility to handle with a radioisotope. So there we cannot use a large centrifuge. And also amount of sample, we have to work in a small scale of sample to somehow. If we use a large, so we have to use a lot of radioisotope and expensive and dangerous or something like that. So small. So separation was not so good as before, but we could separate, A2, A3, A1, A2, A3 band like this. It's a little bit more overlapping, but we could separate. Okay. So after pulse labeling, it's only 1 minute. It's very You have to work so carefully. And then labeling is, this is PS1 reaction center. Was mainly detected in only A2, no smaller fraction. We couldn't detect, we barely detect signal in A3, okay? And also some minor PS1 subunits are also present in A2, not A3, not much, okay? And also this is PS2, as I told you, this is PS2 D1 protein that turn over very rapidly. Was present in not only A2, but also here in the upper phase. So it's not properly assembled proteins are present because this is a very strongly labeled turning over. So signal is so strong. So this is LHC2, okay? And after chase experiment, it's difficult to say, but it's this PS1 protein shifted to A3 mostly. You know? Separation was so delicate, but it shifted. If you look at the PS2, signal is here, peak is here. No, the peak of PS2 and the PS1 was the same here, but this is a little bit shifted. But there's a strong signal here, but this is sometimes aggregation of PS2 is migrated like this. So, but still, some are still remaining here, but it's shifted, obviously. Okay, right? Here, no minor PS1 subunit is almost missing. It's shifted. LHC2 is more abundant because it took some time to integrate into the thyroid membrane because it translated in the cytosol, translocated into thyroid chloroplast, and then translocated to the thyroid membrane. It took some time, so it delayed. So, okay, this is the S3 band. So PS1 subcomplex detected in H2 is assembly intermediate. So it's not artifact. It's assembly intermediate. So we couldn't detect much assembly intermediate, but only assembly intermediate of PS1 core lacking LHC1 and PSAG and PSAK was right here. This here. And then this is H2. Then H3, LHC2, LHC1 is associated, and PSAG and K associated to fully assembled PS1. You see, this is consistent with here, G and K are lacking, deficient. And LHC1 is very low. So it's before the LHC1 and PSAG and K assembled PS1 core subcomplex accumulate. So this is only assembly intermediate we could detect. Because this step, the following steps, this following step is some rate limiting slow. So transiently, this subcomplex accumulate as a detectable level. Okay. So that's it. This is the same as greening experiment, right? PS1 is first accumulated A2 band. So this independent experiment indicate the same lead us to the same conclusion. So probably this data can be reliable. Okay, so probably I stop my talk here and then ask you the question. I told the previous lecture that look at the data very carefully and speculate what we can extract from. So you don't have much time to see the data, so it's difficult for you to say something from data, but just by asking and hearing an answer, your thinking will be activated. So it's important not to listen to the lecture quietly, but asking even a stupid question is okay. Just talking stimulate your thinking. Okay, so I stop my talk and then ask you some question and comments or welcome, and then is that okay? Okay, Sam, I ask you, how do you think it's the greening? There's some the derivative of chlorophyll, intermediate, accumulate. Do you have any question about this? No. What happened to the plant or algae if accumulate is not chlorophyll, not only chlorophyll A and B, intermediate accumulates? Gives you any questions? Maybe. No, this is a hint to ask a question.
The higher level of intermediates, they may be.
Sorry, say.
Higher level of intermediates of this pathway, they may be toxic to the plant.
That
they may be toxic to the plant.
Toxic plants. Okay, the question comes. So this accumulation, if you think that this intermediate are not functional, has no proper function as chlorophylls, that could be toxic under illumination, right? That question you didn't have? Okay. So far we don't know, but I'm going to talk later about this mutant that accumulate only geranyl chlorophylls. That analysis of this mutant gives us answer.
And are those steps compulsory? Without those steps also can we get chlorophyll?
Sorry.
Are these 3 steps compulsory, or if one step is missing in between those 6, will we get the?
If the GGR is missing, so the no chlorophyll A and chlorophyll B accumulate.
If we knock it off, or if
knock out, yeah, of course. This process is blocked, so only chlorophyll A, geranyl chlorophyll A, and probably geranyl chlorophyll B accumulate, but no other. That is the case. So maybe you may have a question that geranyl chlorophyll A, does this geranyl chlorophyll A or B functions to collect light energy? Or in the case of reaction center, the chlorophyll A is involved in the electron transfer, charge separation. So geranyl chlorophyll A whether it acts as electron transfer component, primary electron donor or primary electron acceptor, redox reaction happens with this geranyl chlorophyll? That is the question, right? Another question is probably the granular chlorophyll A has 3 additional double bond here compared to chlorophyll, the phyto. No, this is only here. Double bond is here. So double bond mean this double bond is not flexible, right? So shape is rather fixed, but phyto is more flexible, right? So if these chlorophylls accommodate into chlorophyll protein complexes inside the proteins, right? So this is very like a rod, it's rigid. This is very flexible. So to accommodate into the proteins, I think this should have advantage. No, it's if you accommodate in some specific place, and this part disturbs the proper binding, then this can be bent or form flexibly, but this doesn't. So that prevent the proper binding. That's possible, right? You can speculate. So, but somehow the answer is there. Chlorophyll protein complexes are present. That is one of the reasons why we did this experiment is that granular chlorophyll A prevent the proper assembly of chlorophyll protein complex or not. It seems to be working. So chlorophyll protein complexes structure is rather flexible somehow. More modification of the structure of pigments did not affect assembly. So the function, how about function? That is we discuss later, not today. So just keep it interested.
I have one question there. So GGR, it is involved in the vitamin K tocopherol biosynthesis also.
Yeah.
So what happens in the mixture? That also get affected or?
Since the detection of the vitamin K phytoquinone was difficult, so this time we didn't analyze carefully. Because to be honest, from the beginning of our experiment, we didn't think about others. We just focused on only chlorophylls. And then we noticed, but it was a little bit too late because the student working this was graduated. So, but PS1 is assembled anyway. So, okay. So other questions? Okay. It's always have questions. But say it again.
These different chemical reactions are happening in same place or in different distributed because they are using same enzymes.
GGR, yeah, these are
same enzyme.
GGR, granule reductase.
Synthesis also in localized in same place or in different place.
GGR.
Yeah.
We haven't tested, but at least a GGR, granule reductase, is present on the thyroid membrane.
So other experiment also going on there only. Other alpha token.
These are all very hydrophobic molecules, so these are not present stably in the soluble part. So it should be present associated with the membrane protein or lipid bilayer. So at least a vitamin K1 or vitamin K1 or phyroquinones are associated with PS1 in the middle of the hydrophobic G region. And tocopherol is, I'm not quite sure distribution of tocopherol. Maybe in the membrane. And of course, chlorophylls, all chlorophylls are associated with proteins. No free, practically no free chlorophylls, because that free chlorophyll makes no reactive oxygen species under the light. That's so toxic, so it's very dangerous. So GGR works as I showed that in the reducing granule chlorophyll present in the complex chlorophyll protein complex. So this is consistent that if GGR is present on the thyroid membrane, this is consistent, right? Data. Any other questions? Okay.
So what are the two pathways involved? You told two pathways are involved in also. You told two pathways are two pathways.
Two part of what?
You told there are two pathways involved in this chlorophyll A and chlorophyll B. That cycle you told.
This one? Or part, the same enzyme?
Two pathways.
Two part.
This is one pathway, this is one pathway.
This one pathway? Okay. Both way. This way or before it's associated, granule phosphate is before associate here also. We don't know exactly, we cannot say for the moment. I don't have enough data, evidence that to distinguish this reaction and this reaction. The paper, as a published paper or a review paper, indicate that in parallel these two reactions may occur. But yeah, it's possible that, but we cannot exclude one of the reaction. But probably this is actually occurring, okay? But we cannot distinguish, deny this reaction occur before this reaction. So quite detailed, it's difficult to.
Before slide and this slide, both headings are same. No. This one, no, this one. Next.
Sorry.
Gerani and geranyl both are same or different?
Geranyl reductase, oxidoreductase.
The headings, are they both same? Above?
Geranyl reductase. CHLP, that's it. This is the name of the enzyme, and this is name of the enzyme gene. CHL is involved in the gene involved in the chlorophyll synthesis. So usually biochemists assign the enzyme and name it as a granule reductase or granule oxidoreductase, and then later gene or mutant will be isolated, and then they name later they name that the genes involved in chlorophyll synthesis renamed as CHL, A, B, C, D, E, F, G, H, O, something like that to make things easy to understand. So sometimes naming of enzyme and also naming of the gene or gene product. This is uppercase C it's proteins, and the lowercase c is the gene name, and italic is gene name. So this changed. So sometimes, molecular biologists dominate biochemists, and then they rename, ignoring the enzyme name. So, but sometimes we have to write both. That's more kind. It doesn't say anything. It may be related to chlorophyll synthesis, but GGR is granule reactor, so it's clearly show the function. So it's better to write both for readers. It's very kind. But it's sometimes people don't do that, so I'm a bit confused. Sorry, CHL, but this is name, and this is more recent, more later, named more later than original. This happens many times, so just don't get confused. Always the name of the enzyme and the name of genes and the gene products are not the same, but it's the same, meaning the same. Okay, so now.
Takahashi, I have some questions.
Okay.
Not question.
Not question.
Very nice talk.
Oh, thank you very
Halfway through, I have listened. And a lot of detailed things, so whatever you have done, your research about me. Technically, actually, I am looking for the feasibility of these type of experiment in postgraduate level. So, in that context, what amount of your chlorophyll is required for your radioactive experiment?
The pigment
Because
composition.
you told it's a small.
Pigment composition by HPLC, you need only small amount, about 50 microgram chlorophylls. It's very volume is smaller better, because it's if you want a separation better, you apply the HPLC a small volume. It depends on the loop. For example, 100 microliter or 50 microliter. Yeah, the lipid determination of the lipid composition is, if you have a good HPLC, it's not difficult. But detection system, if you need diode array, if you can measure the absorption spectra at the same time, it's easy to assign the band, because band is not always the same position. Depend on the sample or depend on the solvent. It relatively changes. But if you want to separate protein, also you don't need much. For immunoblotting, you need a very small amount, because Western blotting is now very sensitive if you use ECL detection. So very low level, even lower than pigment analysis. But if you want to separate the chlorophyll protein complexes, it's better to see the band, green band. If you don't see, no, if you're working with this, if you don't see band, very discouraging. But of course, for analysis of protein, you don't need much. But in this case, you need a more sample, 100 or 200 microgram chlorophyll at least. But it's still relatively small amount. It depends on the size of the tubes. So, but from the plants, sometimes it's difficult to get enough samples. The chlamydomonas is a liquid culture. You just need increase the volume of culture. It's easy. But plants, sometimes it depends on the size. For example, Arabidopsis, you have to make many. But chlamydomonas is easy. That's why the chlamyd is advantage. You don't need much space. You can save money and save space, but of course, sometimes we need to know what happened in plants, vascular plants, or very useful plants. Chlamydomonas is not useful, so some people criticize Chlamydomonas is not so. You may eat, but you don't want to eat. So that's. But still, Chlamydomonas is for the basic science, plant physiology, plant biochemistry. This is very rapid.
Yeah,
rapid
and single cell.
Single cell and rapid, so you can grow the cells in a few days. But in the plant, you have to wait weeks, some a month. But it's very fast and quick. So there's a disadvantage and advantage, but it's a basic. Once you analyze the basic phenomena using a chlamydomonas, and then something interesting things that you can apply for use for the plants. That's good. So both. Some people work both Chlamydomonas and Arabidopsis. So that is.
Thank you, sir, for the wonderful talk.
Okay.
It's been informative. If you have any questions, you can.
Yeah, welcome to come to me to ask questions.
In fact, Professor Takahashi was talking about the details of a particular chlorophyll. Okay, yeah. So that's why. So maybe I am not that much of detail, but today we will be talking about because today the responsibility has been given by Dr. Rajgopal sir, that membrane glycolipids in photosynthesis, thylakoid biogenesis, and chloroplast development. So very nice topic it is, and I was not much more aware of this one, but I could see that yes, it's a very interesting things which can be also give you some of the clues as well as some of these your ignitions in your thinking process to come out some of the good topics as well as questions. So if you look into this, the chloroplast, as you know that because last few days you are acquainted that it's a double membrane organelle and called the plastid, and which are unique to the plant and algal system. And perhaps Professor Takahashi was talking about the alga, means all the chlamydomonas we are talking about as a, and they are capable of oxygenic photosynthesis, and of course it has a outer and inner envelope membrane, and chloroplast have extensively developed internal membrane system. That's what we talked in the last lecture, and where the photochemical and electron transport reactions generally takes place. So thousands of proteins and pigment as well as the photosynthetic factors, cofactors are available there, and they are embedded and in the bilayer formed by the glycolipids. So these bilayer membranes, which is a glycolipid in nature, and evolutionary, if you are looking at that chloroplast and cyanobacteria, they have the same lipid composition. So as a result, the non-phosphorus glycolipids are the major constituents of this membrane, and although the typical cell membrane in animals as well as the fungi or non-photosynthetic bacteria use the phospholipids for building blocks of the lipid bilayer. So as a result, what happens, this is a global phenomena, you can say, in a cell where the membranes are there. So, and as you know that there are stroma and grana, and both the outer and inner membrane, and we will be talking about this outer and inner membrane and what is the compositions of this one. If you are looking bit the normal things that what the chlorophyll is doing for, because we are looking for these things here, that in the visible light, because all these phenomena or the chlorophyll activity is in the visible light spectrum, not in the far red or into the infrared region. So as a result, what happens that the visible part of the spectrum, it has the violet red color, and more or less all these chlorophyll molecules or the pigment molecules, they absorb these lights in different wavelength, and of course we are looking for that the green plant, because the etiolated plant, how it becomes green. So in that case is green plants nothing but they absorb all the white light and reflected back the green light, so that we are seeing the green things of the leaf. And if you are looking at the chloroplast, because we will be talking about these portions today, so in that case is these have, as you know that the inner membrane have a stacks of your thylakoid stacks, and these thylakoid stacks, they have the membrane. So and these membrane, they have the light harvesting chlorophyll complex, because this chlorophyll complex, as in the previous lecture you have seen that the magnesium, this part is common there, phytol ring is there itself, and all these rings, these hydrocarbon tails, they are very much flexible in nature. But in the same cases, if you are looking into this, so as a result what happens, this long nonpolar hydrophilic tail, this is generally anchors into the lamellar membrane, and head portions has the complex ring with the magnesium at the center, which generally where the light emerges is trapped here. So this is the machinery of the light your trapping system, and these portions is attached to the membrane, so that we can have this chlorophyll molecule, which is generally acting as a molecule which globally produce the foods for us. So this is the normal things where we are looking for that. And if you are looking the absorption spectra, that we have the chlorophyll A, this chlorophyll B, and then we have beta carotene. So this A and B, they have a region of this red region, whereas the blue or green region have the beta carotene, because this beta carotene perhaps act as a molecules where generally scavenge some of these A and B molecules perhaps are protected this one. So as a result, we can see that, because if you are going for a biochemical analysis, we will be having a large number of chlorophyll molecules where chlorophyll A, B, then C1, C2, C3, D, F, and everywhere these heads are there itself, only there are differences in the tail region. And of course, if you are looking for the carotenoids, where the xanthophylls and beta carotenoids are there, and so they are the linear structure, whereas phycobilinoproteins, if you are looking into that, so where the phycoerythrin, phycocyanin, and phycobilins, they are the components, they are able to do the photosynthesis in our spectrum. So if you look at the history of this evolution of this chloroplast, how it comes, because cyanobacteria developed oxygenic photosynthesis about 2.6 to 2.5 billion years ago, very, very long back. And approximately 1.9 billion years ago, after the endosymbiosis, generally that cyanobacteria is placed in the host cell, so that in the evolutionary process, the plastid has emerges in the eukaryotic system. And about 100 to 200 million years ago, red algae and rhodophyta, as well as they have the common ancestry with these previous, and we could see the green algae, because they are perhaps this 100 to 200 million years ago, we could see that the same kind of pigments are there in your red algae as well as brown algae, whereas green algae appears in 1.1 billion years ago. So you start from that 2.6 billion years ago, then come to the 1.1 billion years ago. So what is the long space where the evolution takes place, and the chloroplast has emerges, and that gives us the food right now. But if you are looking back, because I several times told that our age in the earth and the earth ages and the evolution, if you are calculating in terms of Darwin's or mutation, it is not matching. Earth age is quite younger as compared to the human evolution. So as a result, from the single cell of Chlamydomonas to the very big tree, angiosperm, so it is quite astonishing to understand what exactly it is, and sometimes maybe we are implanted here from some other planet, it may be, we do not know. Okay? So if you are looking to the terrestrial plant, so where the carotenoids in the first positions, where generally they are absorbing the green lights, as well as to some extent the blue lights, and then it can percolate it up to the chlorophyll B, then up to the chlorophyll A, where the chlorophyll A and B also can take this red light also can take care by this chlorophyll A and B. So we have a reaction center and chlorophyll A having these both PS1 and PS2, where this P700 and P680 is there. Then marine habitat, if you are looking to that, this brown algae and dinoflagellates. So generally it has also the carotenoids, then chlorophyll C, then chlorophyll A, and also have your PS1 and PS2. Then if you are going for a cyanobacteria, so in that cases fur reds are also absorbing by this and some of the yellow lights also absorbing by the PC, then APC is by the red and fur red, then chlorophyll D is there itself to take care of, because all have the this your magnesium rings are there, and of course chlorophyll D's are there and some other unknown products are there. So this it has also the reaction center, and of course cyanobacteria, when we are looking for that, so during that time the chloroplast structure is not exactly this normal chloroplast structure of this angiospermic plant. So and if you are looking into this another cyanobacteria group, so where the your bilinoproteins are there, then chlorophyll F, then chlorophyll A, and so it is as similar of this one, and then also it has a reaction center to manage both the PS1 and PS2 system. And red algae, say for example, so where also we have because in this process generally we are missing this chlorophyll A part, sometimes chlorophyll B or C or F part also plastocyanins are taken care of, and we have the photosynthesis. So as a result, what happens that all these cases when the chlorophyll A and B and C are there, most of the major chlorophylls are generally chlorophyll A and chlorophyll B, and formula wise they have the differences, and chlorophyll A possesses the methyl groups, whereas the chlorophyll B possesses aldehyde group, and it is essential pigment chlorophyll A for the photosynthesis, whereas the B and the other carotenoids etc., they are called your accessory pigments which absorb the energy of the different wavelengths and transfer it to the resonance or to the chlorophyll A, so that the chlorophyll A is supported by the chlorophyll B or the carotenoids or any other pigments which is there itself in the hierarchy. And of course the chlorophyll A spectrum, if you are looking to that, of course they have a maximum spectrum in near about the 650 to 1700. The red region as well as the blue regions also will be having someone, and chlorophyll B and carotenoids, if you are looking into that, so that is generally in the blue and green as well as the violet portions it is covering. So as a result, what happens, both the chlorophyll A, chlorophyll B and carotenoids, they are having the major role to play there itself, and of course everybody has this ring structures which have the much more similarity with this hemoglobin, where only the iron is replaced by this magnesium, and in the last lecture also we talked that how it is the similar structures is operated there. One is carrying the oxygen, whereas this is for your photosynthesis. So and chemical compositions, if you look into that, proteins we have about 35 to 55%. So that's why I am very much I will be very much happy that after this lecture, if you are taking much more of plant, so you will be getting a lot of vegetables here, and here maybe you will be having this you will be visiting this facility in the top, where they are also producing the proteins, this LSC1 complex and LSC2 complex as a powder, and they are selling in the market. So okay, so these lipids and you will be astonished that they are from even the larva of the any of the larva. Okay, although you are vegetarian, some are vegetarian, but it is larval protein. And sometimes the cabbage, the waste leaf of the cabbage, they are collecting and isolating this LS1 and LS2 complex. So that's why this is the protein which is marketable, and they are going for that one. And lipids, if you are looking into that, because we will be talking about the lipids 20 to 30%, carbohydrate about 3 to 7%. So that's why nowadays a lot of people have the diabetes in our country. So the carbohydrate, very less carbohydrate are there. Only the proteins you have much more. Chlorophyll about 9%, so dry weight, and chlorophyll A, out of that total chlorophyll of 9%, about 75% are chlorophyll A, and chlorophyll B is 25%. So chlorophyll A is much more there. And of course, to support that one, carotenoids we have 4.5% and nucleic acid 3 to 4% in the normal positions. So this slide is quite vital for today's lecture. The major glycolipids in the thylakoid membrane, if you are looking into that, yes, they are very big names, but later on we will be accustomed with this. So MGDG, then DGDG, SQDG, and PG, these are the 4 things. They are much more vital glycolipids in the thylakoid membrane. And if you look into this, that MGDG, it is mono, it is di, and it is sulfino, a sulfur containing one, so that SQDG, so they are your glycolipids, whereas phospholipids is your PG is your phospholipids. And then if you are looking into these uncharged and negatively charged anionic proteins, so they are, these two are your SQDG as well as PG. They are your negatively charged or anionic proteins, whereas uncharged proteins are MGDG and DGDG. And non-bilayer forming is your MGDG, whereas bilayer forming, all the 3 are bilayer forming. So as a result, you can see that these are the 4 lipids, the glycolipids, those are the major component of the cell membrane, which is going for that. So your MGDG and your DGDG, because all these terms will be coming very frequently in the next slides. So, and if you are looking the membrane compositions of the plant plastid and cyanobacteria, thylakoids, MGDG, we have about 53%, so whereas DGDG, we have 23%. So monolipid, dilipid is very less there itself, inner envelope 49% and 30%, and SQDG 5% only, PG 8%, outer envelope about 17% of mono, di is 29%, and your SQDG is 6%, and non-green plastids, those are there itself, because sometimes that's just few minutes back, the lecture was that how this non-green plastid become green, and etolated plastid can be green also. And I just before the introduction, I was asking to Prasad that in the seed itself, the embryo which is there itself, it has a cotyledon, and that is not green color. Then there may be some plastids are there, non-green plastids are there, and during that time, whether all the enzymes, whatever you have talked about in the intermediate enzymes, they are there itself, they are activated or non-activated condition, or after the putting of this plant in a suitable environment, they are activated in the nuclear genome, and then this has a crosstalk into the chloroplast or leucoplast, and then become chloroplast. What is your answer? Because in dormant conditions, the plant having the cotyledon, which do not have any chlorophyll, so must have non-green plastid there, and during that time, all the greening process, whatever the enzymes are there, so all they are present there itself, and then it is finally activated, or there is a mechanism of transcriptional regulation which can trigger after the environment suits to germinate that plant, and then the etoplast or leucoplast become chloroplast, and during that time whether the chlorophyll is synthesized or not. So these are the questions, very basic questions, and you have to answer. We are old enough, we are not able to give answer. Only you are the researcher, you can give the answer to us. But anyhow, so that's why you can see that there are variations of, because major portion of this your MGDG is much more in the thylakoid, and then after this outer envelope very less amount, whereas this DGDG is more or less same type, and whether the MGDG is converted to DGDG or not, that is a question, and or it is converted to SQDG or not, or all these three are essential for not. So in that experiment, because professor was talking about that they will be having some other talk, that meta-analysis, because if you are silencing some of the genes or knock out some of the genes, and then only you can have the effect that what type of your deficiency they have. So as a back, how it could be this one, yeah. So if you, because to give a normal idea, theoretical idea about the PS1, your complex, because this is a computer generated complex of side view and top view, where the monogalactosidal diacyl glycerol or MGDG is present. So these colors, where it is there itself, they are MGDG. Then DGDG is digalactosidal diacyl glycerol, so they are your this color, and all these red color balls, small red color balls, they are oxygen molecules which is attached there. And then PG is also there itself in this blue color, this SQDG is in blue color, and green color we have PG. So these complex, all these glycoproteins, they are embedded into the structure itself, so that a PS1 systems can produce or in PS2 systems where the dimer formation takes places. So it's a monomer and then dimer formation takes places. In that cases, we will be having a lipid molecules where 20 lipid molecules are there, and out of the 20 lipid molecules, 6 MDDGs and 5 DGDGs and 4 SQDGs and 5 PGs form this complex. So in that complex, what happens, the polar head groups of all the SQDG and PG molecules faces the cytoplasmic site of, whereas those of the MGDG and DGDG molecules, except one molecule, they face into the luminar side. So that means topologically, these attachment is quite interesting to see, because you have to visualize the things, so what it is going on there itself, not theoretically all these 3-4 terms. So in that cases, and of course, if you are looking the locations of these 3 PG molecules, so there generally we have the PG664, PG649, PG702 located near the QA binding site of the PS2. So in that cases, generally the oxygen, phosphorus and carbon atoms shown in the red, purple as well as green color respectively. So in that cases, what happens that we have different molecules, and D1 protein, D2 proteins are also there. It is in a specialized folding. So mostly it is of alpha folding, and then in the locations of 2PG, if you are looking into that, so there are also 3 protein molecules located near the QBB binding sites of the PS2. So as a whole, you can have a things that because we have talked about the chlorophyll, then inside the chlorophyll the different components are there, and in PG experiment generally we used to do anthocyanin or carotene and chlorophyll. But inside the chlorophyll also there are a lot of things to do, and also HPLC can give you the better solutions of each and individual component. And component wise, when you will be going for any of the experiment to do, so in that cases you need 2-3 things very important. Mostly those are doing the MSc, at least I am telling for them, that to learn the SDS page, to learn the isolation of the chloroplast, to learn the spectrophotometry, where this fluorescent your detector should be there, so that you can analyze the peaks, very small peaks also, and what exactly it is. And to get that one, of course, you have to need to take the help of the biochemist as well as chemistry people, that these small peaks of the compounds, what is the structure and how it is assembled with these. That's why in this course I think there are some computational biology which is also involved there, and which is very much lacking in the UGC syllabus, because as I know that all the UGC syllabus do not have much more of this one, at least for the botany people. Life science people, biotechnology, they have bit. Okay, but anyhow, so in these cases, so glycolipids in the thylakoid membrane of the spinach, if you are looking into that, so chloroplast thylakoids, this bar is your chloroplast thylakoid, then chloroplast inner envelope, and there are a lot of variations there where the thylakoids have the much more of about 55% of the chloroplast, whereas inner chloroplast they have a variations there itself. So the variations of the MGDG, DGDG and SQDG there we could see, and variations generally in the mono we have lot of variations here. DGDG do not have much more variation, it's a constant one, whereas SQDG also not that much of significance. Of course, the PC has again a variations there. So plastids, that's why have a specific role in support the plant growth of life cycle that involves the synthesis of the lipids, amino acids, purines and pyrimidine bases, and various hormones and secondary metabolites, as well as the nitrogen and sulfur. So you see this particular membrane structure, they have lot of functions to do in the chloroplast, and chloroplast membrane is unique lipid compositions with non-phosphorus mono and galactosidyl diacyl glycerol, so MDDG and DGDG respectively, and about 50% of MGDG and 25% of the DGDG of the total thylakoid lipid membranes we could see, and rest are comprised of anionic glycerol lipids and sulfo SQDG as well as the PG. Generally we have after this, out of this 50% of the MGDG or 25% of the DGG, rest are your SQDG and PG, and it's a, so that's why, and also with a marginal proportions of the your phospho hydrodidyl cytol, it is there itself. So, and also the glycolipids in the main constituents of the envelope of the chloroplast and non-photosynthetic plastids, with glycolipids about near about 60% of the total lipids we could have. And also these glycolipids and your SQDG are the minor constituents and are absent in the extra your plastidic membranes of the your plant cells, and chloroplast and cyanobacteria reflect the same origin. Because of that, the common ancestry of these particular components are there both in the higher plant as well as the lower groups. Of course, the pathway of biosynthesis is different, and these highly conserved lipid compositions in the among the chloroplast and cyanobacteria suggest a special requirement of these lipids for the oxygenic photosynthesis. And of course, there are glycolipids. That's why that in the thylakoid, glycolipids plays an essential role in providing a lipid bilayer matrix embedded with photosynthesis protein and cofactor complex. So that's why the assembly of these cofactors as well as the other components is quite important to have, not only the synthesis of these things. And coordinated biosynthesis of the thylakoid lipids with the chlorophylls and photosynthetic proteins during the chloroplast biogenesis. That's why it is very much essential to construct the thylakoid membranes network, which is generally so that we can have an efficient photosynthesis system. So that's why we could see that because this how these lipids generally help in the thylakoid biogenesis system. So in these cases, in the thylakoid glycolipids that provide the lipid bilayer matrix as well as the protein cofactors generally forming these photosynthetic electron transfer systems and modifications of the lipid compositions if you have. So that strongly affect the structures of the thylakoid membrane. So as a result, what happens, the thylakoid membrane, if it is lost or if it is not in a proper stage or in the developmental positions, also thylakoid membrane productions is a very vital essential thing, so where the glycolipids plays a very big role. And crucial lack of the MGDG and DGDG in Arabidopsis cases generally causes no or severe underdeveloped internal membrane structure in the leaf plastid. So as a result, what happens that if you are divided of or if you have a mutant, so in that cases we could see that there are not much more changes, but the partial deficiency of the MGDG resulted in decreased amount of the thylakoid membrane. So as a result, stacking of the membrane can lose, and DGDG contributes a membrane stacking via the formation of hydrogen bond between the polar heads of the adjacent bilayers, and MGDG has cone-like shapes because it has a special shape which has a small head group and which allows it to form an inverted hexagonal phases, where the DGDG has a more cylindrical shape. So in that case, the protein shape and structure, 3D structure of the protein is also varying because of the component, and the ratio of this bilayer forming DGDG and non-bilayer forming MGDG affects the property of the chloroplast membrane by affecting the lipid bilayer for the hexagonal to the laminar phase. So as a result, hexagonal structure of this protein is collapsed because of this if there is a variations in the DGDG and MGDG balances. So it is tightly regulated in the response of the various stresses, because when we are putting lot of stresses during that time, there is a changes in the DGDG and MGDG level. So as a result, that affects the size of as well as the protein structures of the chloroplast, so that the functionality of the chloroplast remain straight. As a result, you generally in normal cases we could see that decrease of chlorophyll content in normal cases we could see, but internally we have the changes in the MGDG and DGDG percentage. And also if you look into this, that acidic lipids with negative charges in their head groups PG and SQDG also affect the organizations of the thylakoid membrane, and PG has a crucial role to in the proper thylakoid biogenesis, and deficiency of the chloroplast PG in the Arabidopsis we could see that that formations of the vesicles, enlarged vacuolated structures generally we could see, so that the stacking of this your thylakoid membrane generally disturbed, and or we can see that underdeveloped when the plant is growing during that time, underdeveloped membranes generally we could see because of this deficiency of the PG in the chloroplast level. So that's why in that previous slides we have seen that there are variations of the content of the PG level there itself, and in ratio of this bilayer forming DG in non-bilayer forming MGDG affects the properties of the chloroplast membrane by altering the lipid bilayer from the hexagonal to your lamellar phases, that also we have talked just in the previous slide. And also it is tightly regulated in response of various stresses, that is also there, and also your deficiency of the SQDG causes the strong curling of the thylakoid membrane. So, as a result, the lamellar structure of this thylakoid generally affects and generally grana stacking have little effect, but in the Chlamydomonas, but in the higher plants we have that type of problems in the SQDG deficiency. And lack of SQDG biosynthesis in the Arabidopsis had no effect in the thylakoid architecture. Sometimes we could see that type of phenomena also. Some of the papers they have shown. And loss of SQDG together with a small reduction in the PG content decrease the grana stacking in the leaf chloroplast. So that means that SQDG and PG they have a correlations between, so that that generally affect individually may not affect, but combinedly they have the effect in the grana stacking. And thylakoid lipids that's why are required for the structures of and functions of the photosynthetic complex, besides providing a bilayer matrix. And X-ray crystallography of cyanobacterial PS1 and PS2 revealed the both complex involved in the thylakoid lipids in their crystal structures with MGDG and PG near the reaction center of both the cases. And PG deficient mutant in the cyanobacterium, we could see that Synocystis PCC 6803 demonstrated that PG is essential for the PS2 electron transport from primary plastoquinone QA to secondary QB. So in that cases, electron transfer is severely hampered because of this PG deficiency. So that's why PG also a equally important part of this lipid biosynthesis process. And also the stabilization of oxygen evolving complex by the extrinsic proteins and terminations of this PS1 complex also it has a effect of this PG, and PG is required for the chloroplast development and photosynthesis, and which is generally found in this Arabidopsis as well as Chlamydomonas mutant. And another anionic thylakoid lipid, that SQDG, is crucial for the activity of the PS2 at both donor and acceptor site in the Chlamydomonas and your Synocystis, which is generally experimented there itself, and it does not have much more effect in the PS1 activity. So SQDG also is not required for the photosynthesis of biogenesis in the Arabidopsis under the nutrient sufficient condition, where the PI is much more, and this is required for the maintaining of the amount of the total amino acids in the chloroplast when the PG content is decreased under the phosphate deficient conditions. Generally we could see that kind of situations. So requirement of SQDG for the photosynthesis differs among the organisms because of that P deficient condition, phosphate deficient conditions, and in the normal conditions we could have different type of effects. And of course DGDG deficient mutant, generally DGDA. So in that cases this is the gene which is responsible for that one of the Synocystis species, shows that DGDG is required for the PS2 activity by binding the extrinsic protein that stabilize the oxygen evolving complex. So that the complex, the binding of this oxygen evolving complex as well as the protein, where the DGDG plays a very big role there itself. And of course mutations of that DGDA gene which is generally sensitive for the photoinhibitions under the high temperature, high light, and repair cycles of the photoinhibitions more severely affected in that cases. So what happens that if you are having a stress of high light or high temperature, and during that time, so this your DGDG generally got affected, and so that we can have a differences. And we could see in this forum, perhaps it is very much not directly related, but indirectly related for our global population right now, because in this state or major state of the India, mostly southern part, they are taking the rice, and we are expecting about 663 ppm of carbon dioxide in 2050. So after 25 years, we will be expecting that one. And temperature rising will be about 3 degrees more in the natural condition. So whatever the temperature it is there, it will be rising. So in that cases, we were just given a task by the central government that what type of paddy can be grown in that high carbon dioxide concentration as well as high your temperature. We just screen about 63 varieties of rice, and out of them we could see that there are, because of course we have not done this chlorophyll part, only we are concentrating in the rubisco part, and we could see that only 2 varieties of the rice, they have sustained this rubisco and they have very good activity, at least up to the 3 degree temperature, higher temperature, as well as your so you can imagine that in our country, if you have that type of scenario, that only 2 variety will be performing after 25 years. During that time, I will not be there, you will be there, and your child will be there. Okay, you will see that. So only 2 variety will be performing, and the rest variety is not performing. What they are doing? Because the photosynthesis when it is, so it is actually converting your photosynthetes or the glucose to the starch, so that we could see that in higher carbon dioxide concentration and high temperature, the biomass is very much increased, but the grain filling, it is not happening. So that's why we have chappy rice. So that's rubisco is not working properly. So that's a less conversion of that one, and of course there are some other genes which is responsible for that. And so in that cases, it is the best forum. You try to practice rice leaf to eat, because there is the protein and everything is available. Instead of rice starch, you just go for your chewing this rice leaf, so that you will be getting the food as well as anything, just like the cow. So in that cases, you have to take the help of this your biotechnology people to isolate the protein, and then take the protein in a liquid or in a glass of water, like that. But anyhow, so that's why they have, because sometimes it is a very, because the till date whatever the variety we have released by this ICR in the breeding program, so not a single variety is trustworthy for giving this one, and we could see that there are 2 your LSC genes are responsible for there, and which is generally available there itself for transporting the silicon from the soil. The silicon they are transporting and keeping into the rice leaf, and this transporter gene, they have changed there itself just like a maize. In this particular 2 variety, those are surviving with your high your rubisco. We do not know yet what is the connections between the rubisco and your transporter gene of this your what you call silicon. Anyhow, so, and also if you are looking for that, DGDG also plays a role in decreasing photoinhibitions under the high light, because if there is a high light conditions during that time, of course this photoinhibition takes places, and DGDG also require for the terminations of the LHC2 complex and accumulations of the galactosidal diacyl glycerol in the DGDG deficient mutant could not fully restore the PS2 functionality, because the trimerations of these LSC2 complex generally hampers So as a result, galactose moiety of this DGDG may be important for the increase with the photosynthetic protein complex of DGDG. So as a result, what happens? We could have that conditions, and 40% of the decrease in this MGDG in Arabidopsis mutant has no effect in the intrinsic PS2 activity, but impaired thylakoid membranes are generally found, and loss of MGDG decrease the level of the cytochrome B6F complex. So as a result, MGDG also have a big role to play into this cytochrome B6F complex, and it is blocked the intersystem electron transfer in the tobacco, so which is generally also found here itself, because in the OGIP analysis also we could see that there are blockages sometimes in different protein complex, and about 80% of the loss of the MGDG in the partial reductions of the DGDG strongly impair the PS2 photochemical reactions, so it disrupts the coupling between the reaction centers and antenna complex. So as a result, what happens that it has a major role to play. MGDG also has a major role to play in the coupling reactions of these reaction center and antenna complex molecules, so that the effective electron transfer is not proper because of the loss of MGDG in the membrane. And it is also synthesized because MGDG is synthesized by the MGDG synthetase enzyme, which is transfers the galactose from the UDP galactose to the diacetoglycerol in the plastid envelope, and out of this MGDG synthetase there are some homologous genes which are responsible for that, and they have also the different effect in that, because generally the MGDG1 is have the major role in the galactolipid synthesis, whereas 2 and 3 are localized in the outer envelope membrane, constitute of alternative pathway for the P limit condition. So when the P limit conditions are there, or deficiency of phosphorus, so during that time they will be taken care of instead of MGDG1 gene, and being a binding block of this chloroplast membrane, generally MGDG is used for the DGDG biosynthesis as a substrate there, and so this is the whole scheme of the productions of PG, then TGPG, then your because this is another path where also the SQDG also can be produced. So as a result, this total biosynthetic pathway, if you are looking into that, there are several genes which are responsible for each and individual steps, and where mostly the block the what you call your things bolts letters, they are nothing but the enzymes which is responsible for that one, and in that cases DGDG biosynthesis is primarily catalyzed by the DGDG synthetase, which transfer the second galactose to the MGDG for the EDP galactose in the plastid outer membrane. So this process generally this DG1 and DG2 found in the Arabidopsis or in other angiosperms, and DGD1 catalyzes the bulk DGDG synthesis in the chloroplast inhibitions with the MGDG1, and DGDG2 functions in alternative pathway of the MGDG2 and MGDG3. So as a result, what happens that the DGDG biosynthesis generally also get affected if there are some changes in these components there itself. And another enzyme also sensitive for the freezing 2, that SFR2, that is endemically different from the DGDG synthetase, which functions from that DGDG and oligogalactolipids. So it is located into this SFR2 enzyme is located in the plastid envelope membranes and transfer the galactose head group from MGDG to a second MGDG or to a oligogalactolipid that increase the number of galactose moiety into the polar head. So, and it does not contribute significantly in the disease synthesis during the normal growth, but is required for the freezing tolerance to absorbed by this plant. So during that time also this SRF2 are responsible for managing whole the process of chlorophyll assembling. And also this in this process the PG synthesis when takes place in the plastid and mitochondria and endoplasmic reticulum in the plant, and the first step of the PG biosynthesis conversion is phosphoric acid into the CDP diglycerol by the CDP diglycerol synthetase enzyme, and 5 genes are responsible for that. In that case, CDS1 and CDS5 encode the functional CDs in the Arabidopsis genome, whereas 1, 2, and 3 are targeted for the to the endoplasmic membrane, endoplasmic reticulum, and CDS4 and 5 are located in the plastid and functions for this thylakoid membrane biogenesis. So as a result, what happens that we could see that also there are functions of this PG synthesis in the membrane also, and the last one is your SQDG synthesis in the plant. If you are looking to that EDP glucose is synthesized from the glucose 1-phosphate and UTP by the UTP glucose phosphorylase 3 in the plastid. So EDP your SQD synthetase converts the EDP glucose to the EDP SQ with the sulfide. So as a result, finally you could see that this SQDG generally increases in the response of the P deficiency system in parallel with the decreased PG content, which may help in the proper balance of anionic charges thylakoid membranes protein of this one. So as a result, if you look into this, some of the results we have taken from some of the papers that these chlorophyll accumulations in the PGP1, 2 with the P deficiency, P1 deficiency, that we could see that there are changes in the chlorophyll A, chlorophyll B, and chlorophyll A by B ratio. So of this wild as well as the mutant, and also morphology and diameter of the plastid nucleoids of the protoplast from the wild type as well as PGP1, 2 levels under the PI and non-PI conditions. Generally, we could see that there are nucleoid formations, which is these small fluorescent particles correspond to the plastid nucleoids in addition to the large fluorescence from the nuclei. And we could see that there are variations in the nuclear diameter also. So as a result, what happens that conditions of your different glycolipids also can leads to the formations of different size of your nucleoids into the chloroplast, we could see, and that has been supported with these because in the wild type, if you are looking to that, because this is Kobayashi et al. It is 1914. So Plant Cell Report, they have reported that this wild type, they have D1 enhance D1, then D2 also enhance, LHCP complex also enhance there itself. But in the mutant, we could see that there are P plus and P minus mutant. We have the variations there itself. So less amount or even nil amount of this D1 and D2 protein as well as LHC complex. So that's why we could see that because all these theoretical aspects what we have talked about, that can be also experimented, and also these are all culmination of different papers. I really do not work that much in that particular subject. And also if you are looking to the thylakoid lipid synthesis in regulation of the chloroplast biogenesis, that generally the photoreactive chlorophyll allows the plant to convert the light energy into the sun. It is normally for the chemical energy, and most of the cases MDDG plays a vital role in the xanthophyll cycle by the solubilization of bioxanthin and mediating the binding of this bioxanthin to the epoxy side to the thylakoid membrane, so that the hexagonal lipid structures formed by the MGDG in the thylakoid membrane is essential for de-epoxidation and of the xanthophylls, and therefore involves the photoprotective and non-photoprotective photochemicals quenching of the photosynthesis. So, and also we could see that MGDG biosynthesis is essential for the thylakoid membrane biogenesis, and internal membranes of this biogenesis under the P deficiency condition includes the chlorophyll accumulations and partial formations of PS2 complex in the MGD1 to genes. Absence of this and photosynthetic electron transfer was still desulfonylated in the case of the mutant. So, thus the lipid biosynthesis is essential for the nucleoid modifications during the thylakoid membrane formation, as well as biogenesis and expressions of photosynthesis associated genes in the nucleus, as well as the chloroplast and or the plastid, and also that regulated structures of these DNA and RNA polymerase complex, because that GUN1 gene is generally found which is act via the nucleus, and generally for the thylakoid biogenesis that affects the expressions of the photosynthesis associated nuclear genes at the onset of the chloroplast biogenesis through the plastid signaling pathway, which could be independent of the photosynthetic activity. Because of that, MGDG12 and PGP12 shows no photosynthetic electron transfer activity under the P deficient condition. So, as a result, what happens that gene which is acted, and maybe some transcription factors which is regulated the genomic constituent, and also the intertalk between this nuclear genome as well as the plastid genome to regulate this lipid synthesis pathway. So, as a summary, if you are looking to that, more or less it is 110. Okay. So, the concerted synthesis of the assembly of the membrane lipid and protein pigments and cofactors are required for the efficient photosynthetic system and chloroplast biogenesis, of course, and thylakoid lipid biosynthesis can trigger the expressions of the photosynthesis associated genes in both the nucleus and plastid, and fatty acid synthesis may be linked to the plastid protein synthesis, and so that the biogenesis of the thylakoid lipids is coordinated with the biosynthesis of the photosynthetic protein cofactor complex at the transcription level and post-transcriptional level in response to the endogenous and environmental causes. And of course, the reciprocal coordinations of the synthesis also there. Because of that, when some of the stresses become activated during that time, they are coping with the system with the help of some of the bypass route, which is also we could see. So, that's why in a normal case, this is perhaps the last slide. So, where if you are maintaining this chlorophyll A and B ratio, because if you are going for a engineering of any of the plant where the AB ratio, if you change, so in that cases you can have a plant where much more of your light harvesting system as compared to the normal one. So, in the normal one, if you are interested for having much more of productivity, of course, grain filling, say for example, is a concern. So, where we need to have entrap large number of your energy, and chlorophyll is one of the major component where the glycolipids are playing a vital role for stability of the membrane, as well as the LS1 and LS2 complex, as well as other coenzymes which
need to be activated and of course well thought, so that we can have, if we are engineering some of the plants having the chlorophyll A by B ratio, where it can give much more productivity. And thank you very much for your patience hearing, and perhaps you are late for 10 minutes for lunch. Any questions if you have, or I am leaving this slide to you, it is up to you, and anything. If any questions, I will be glad to answer anything if it is. জ্যোতি সেটা পানি আছে?
Sir, I have one question, sir.
Sure.
From dormancy to germination.
Yeah.
Thing that you told is very interesting. From etioplast to chloroplast.
Yeah, chloroplast. Yeah.
What I think is all the genes, they are already there
In the system. Yeah.
under favorable conditions, the transcription machinery or protein degradation process, activate those genes, chlorophyll biosynthesis.
Yeah. Yeah.
That is how chlorophyll is synthesized.
Yeah.
During after the germination.
Yeah.
Am I right, or is there any?
No, that may be the cause, because sometimes also you could see that if the chlorophyll also there itself, sometimes it become also leucoplast. Chloroplast become also leucoplast. Say for example, in a plant, the upper part is chloroplast, lower part is your leucoplast. Root have the leucoplast. So there is a transition where the stem and root transitions are there. There may be some clue anybody can work because some machinery is there, how it is switching on this leucoplast to chloroplast. Or say for example potato, if you are keeping outside, sometimes it is greening on the upper part. So how the leucoplast become greening? So this is some of the things which need to be, because I always told my student that you just go out from your this black and white writing, just go out from there and you think the nature, what it is going on, and take some of the questions, pick up, then digest. If it is not, then go to your sir, because sir is nothing but the gap between the two words of a sentence. One sentence, many words. If there is no any gap, you cannot understand anything. The gap is your sir, and gap is also answer as well as question. So if you are thinking much more, you will be getting much more, and always you try to think three-dimensionally. What exactly it is going on? So bilayer membrane we are talking about. In photograph, it is only a single layer, but 3D, what it is going on. Now the computational science, they are giving much more of your focus, and you can get much more of understanding process. And that's why sir was talking about that it is our duty to ignite. Out of these 30 students, if one is ignite, this is more than enough for us for this lecture. Thank you very much. Once more, thank you very much. And I profusely thank to Professor Rajgopal sir, as well as all the participants, as well as our Jyoti and sir also, that to give this opportunity. And yesterday actually I could not make it because of that. We have a crucial problem in the university that in the +3 level, we required a question day after tomorrow, and 10,000 students are halted. So the message has come from the Vice Chancellor that you just prepare the things immediately you send. So yesterday night I just prepared that one, sent that one, and then I came here, and tomorrow maybe I have to leave to Bhubaneswar. So thank you very much, and wish you all the best for this fruitful your training, as well as future of your everybody's life. Thank you very much, and once again thank you, sir, for nice meeting you. And maybe sometimes if we have actually, if we could know earlier, so I could connect you to take to our university, and the next time when you'll be coming, it is our wholehearted invitation that you should visit us. Okay. Thank you very much. Thank you very much. Thank you very much. Thank you very much.
Thank you, sir, for the wonderful talk. It is very informative. We could able to know all the lipid components.
Yeah. Yes. 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 Yes.
With this,
Yes.
we conclude this session. So all of you, please join for the lunch. If you have any clarification, first one, can you ask them to sit in the front row? It's okay.
Is it audible? Fine. So good afternoon one and all. Myself Mohammad Yusuf Jamal. I did my PhD with Professor Rajagopal sir. This year I got awarded, and so here today I'll be talking and having a hands-on training session with all of you on omics analysis, and omics analysis particularly with respect of metabolites and metabolomics. So you can take in your future, if you require, you can take proteins also and RNAs also, not a problem. Like whatever the omics analysis is there, it will be having an identity and a value. Okay, so how I would be telling you, all of you, like how you can put that data in a like a publication format. Okay, so like because omics has a lot of you need to quite intricate data is there, and you need to analyze, and some networking analysis and all those will be there. Like you need to see protein interaction or metabolite interaction, whether it is so in that way we'll see throughout this session. And I'll try to, and also you try to fasten. I hope all of you know how to operate the computer system, and also you have a good command of it. And if any doubt or anything is there, please do not hesitate from a 0 point. Okay, don't be in a like you should not ask or something. Please be very I mean you can, don't hesitate to me asking me. Okay, so is it fine? Okay, otherwise Gmail it was coming, man. I was saying.
This thing is also not.
Yeah. Okay. So that time, I think that time you were saying it is coming. Okay.
It should also come here. It's not.
So that one you can screen here, na? That okay. Okay, understood.
That was for data to take me, for me to take and put it up in the lab.
Okay.
For here you need to connect.
Okay.
Device setting, system setting is not coming, like where extend display. Not coming actually. Coming this.
That should be there. Okay, so hope all of you have installed R program and Cytoscape, right? And also I forwarded a zip file of it. So you extracted the files and data from there. It is a Cytoscape, and then are some commands are there in that. Okay, some. Okay, so one more thing, all of you, can I install some software here if possible? Huh? But it will take time, just for if you try to connect, but it is just for showing them. I'll be. Oh, I don't know. Huh?
तो अगर इंडिविजुअली कोई किया तो जरूर
टच पे नहीं है क्या?
अगर इंडिविजुअली कोई किया हुआ डाउनलोड तो उसे फाइल की
नहीं
क्या है
इसको ब्राउजर ओपन करो, काम नहीं कर रहा है. सलीम को कॉल करें. So in that there is a packages in R program. All of you please open R program. So there are four windows you will find. Open R program. You downloaded R and R Studio also, both of it, and then install R Studio. You can search and then open it. Now all of you can come front, okay, so that like if no, not like R Studio. Open R Studio. This is our program. Yes, yes, yes. This is the one. Like this. Enlarge it. Yeah. So in the last corner, there is a one another console is there, right? The very last panel in the bottom. Yes, yes. The very last panel. So in that there are packages, right? Packages are there. So in that you click and then download in search. Yes, yes, yes. Fine. What is it saying? You minimize it. Na this one. What is this? Close it. This one. Yeah. So in packages, na there is a search option, right? In search option, you can download some packages like dplyr, dplyr. If you type dplyr, okay, dplyr and then search. If it is there, no, okay, then go to install packages. Install is there, right? Install. There is one install is there. One column or like, okay, in that install you type dplyr. Okay, so it will show, right? If you are connected to internet, so you can install that. Likewise, you have to download HMISC, HMISC. Yeah, thank you very much. Okay, thank you all of you. Now I have the connection and you can see nicely. Yeah, able to follow? Yes, yes, just follow the screen, it will just fine. Okay, everyone has RStudio by now? Okay, so today we are going to how to put omics data in publication format, hands-on training workshop. So let us go to first of all, there are three softwares which I would like to teach you today, and at the same time how to plot the data. And please follow very calmly. Don't because if you do any mistake, any space or slash error, you will be having error in that. Okay, come on. So it is a long procedure a bit. So just be very patient, and while typing something, you be very careful. Okay. Okay. So let us go to first. I'm going to talk. There are three softwares: R program, Cytoscape, and then we'll go for metabolite analysis like pathway enrichment analysis. What are the pathways in different metabolites we get right? So which pathway they are coming from? Okay, which pathway they are involved? So metabolites. What are the metabolites? Usually, what are the metabolites? They can be anything, or they can be specific molecules. What they can be? Secondary or primary, everything is there. So they can be, what can they be? Any example? Protein is a big, larger molecule. Amino acids, fatty acids. Okay, these are the primary metabolites, and secondary metabolites are quite different, and maybe that get formed because of the some primary reaction between the primary metabolites, that could be because of the enzymes or some other things also. Okay, so please be very interactive. I don't want there should not be any interaction. I should be having interaction. Okay, and fine. Okay, so second thing, so once suppose what we do, we extract the metabolites from a sample, right? From metabolite, from the sample, whatever the sample is, the bacteria, whatever it is, animal sample or what. So you extract the sample at a specific point of time, right? And you do it in duplicates, triplicates, at least 5 replicates or 3 replicates you should do in case of the metabolites. And particularly metabolites are very dynamic species of a living system, right? And within time to time, within a span of time, it changes very rapidly. So because the physiology of an organism goes on, right? So in that way, what happens, so you have to take or extract the metabolites at a very particular time, and at the same time when you are going for the replications, right? So in that case, like sometimes some people just they harvest the cells and then keep it in, just you can freeze it, the samples, but you should not freeze for longer time. Okay, so once we have extracted, I will not be talking about the extraction protocol and all those that. So after extraction, you give it for mass spectrometry or GCMS or any kind of spectrometry methods are there where metabolites get detected, right? What are the like, do you know any techniques for metabolite detection, identification? Suppose you have to find out GCMS is there and LCMS and HPLC. Okay, so like this you can identify the metabolites in a given system. Okay, and then from there you find out that this particular metabolite or array of metabolites has a particular concentration. They are present in this particular concentration. Okay, so when you found the particular concentration, so from there onwards I am going to take over how the you can analyze. Okay, so here we'll be going for metabolites analysis through MetaboAnalyst. Another software is there which is freely available online software. Okay, and you can make use of it, and it is readily accepted in reputed journals. It is not like that it will not be accepted. It is one of the most reputed, like MetaboAnalyst you can also use for your publications. Okay, so second thing, so let us start with cytoscape. So sorry, R programming, and then cytoscope I will tell how the metabolites interact with each other. You can see how the interaction might be happening between the metabolites, among the metabolites, and in the whole network. Okay, so let us start with R program. So why R program is being used here? So as you might have seen, I have shared you one omics analysis data, right? You can see in the screen is not visible. Okay. There it is not coming. Okay. Yeah, I can share it. Okay. Yeah, I have. I checked actually in the after share, right? Just a minute. There is some internet screen. Getting? So all of you open the file and keep it up in a particular folder, okay? But here this one is not coming, I don't know, here. There is screen changes are happening there, okay. Why is this? So take the R program software, open that command, one of the command, and that is like R program. It is not visible. Bye. Yeah, R Studio is there, right? I think R programming one folder I have given in that, right? Jyoti. अरे यार वहाँ चेंज हो रहा है, डिस्प्ले नहीं आ रहा है. वहाँ चेंज हो रहा है लेकिन हाँ हाँ नहीं भी बीच में आ रहा था लेकिन वहाँ पे नहीं है. हाँ आगे. Okay. It's coming there, right? Okay, fine. Okay, I got it. So now you can come and see here on the screen. So I shared you omics data analysis. So I just zipped all the folder which I have open here. And so first we'll go for R programming, okay? So you can open this, okay? And what is the path of this? So if you see, if you click on it, so this is the path of this folder. If you have to come directly from through some software here, directly access this folder, this is the path of this folder, okay? So when we go to R Studio, so first let us, I have given you 2 R programs. Okay, okay. So I'm starting, I have given you 3 folders, cytoscape file, metabo analyst data and R programming, right? So first we'll go through the R programming, okay? So in that R programming, if you click, you find there is one CSV file known as A versus C, okay? A versus C, if we open it, so you can see I have actually I extracted the metabolites, I gave it for GCMS analysis, and from there I have taken out the metabolite concentrations, okay? So this is propionic acid, like I have given some few of the metabolites like this, okay? So this is like propionic acid and all those metabolites, their name is there. I have given a ID, right? Sample ID I have given, like replicates are there, and then at the same time sample name are also there, like that is the condition I have used, and then these are the concentrations, okay? That is what you fetch out completely finally from whatever the software data extraction protocol or methods you use for the metabolite analysis, data gaining, I mean data you get from the software, from the GCMS and all. So in this way it comes out, or you have to somehow format it in this way, okay? So this is another file I have kept, so that all the names of the metabolites will be there, complete full name of the metabolites are there. And sometimes for R programming and not for R programming, for the cytoscape you have to give for 3-letter code for each metabolite, okay, in order to shorten it, and at the same time otherwise it will not jumble upon like so many, it will get colliding, the names will be colliding because the space will not be good. Is it audible? Fine. Okay. So now I have given two programs, hierarchical cluster heat map analysis and making data for the interaction analysis. Okay. So first we'll go for hierarchical cluster heat map analysis. Otherwise, okay, let us go it for the hierarchical cluster heat map analysis. Okay. So what is that hierarchical cluster heat map analysis? So what happens, like each metabolite has a data point. Each metabolite has its concentration. So depending upon the heat map is what? Heat map actually represents the concentration of a metabolite or a molecule, and depending upon the color, you can say that color gradation, there will be some color gradation or some R and P value, there will be so many things I will tell. So in that you can represent the metabolite concentration directly or its appearance, okay, and how is the concentration of it. And so let us make it right somewhat like this gradation. This is also a heat map, right? Okay. So when we open R, here, just see. So you can open directly if you click. If you have already installed, you can directly click on it. It will open in R program. Okay. So these are the here some packages you need to install. R is very easy to use, and it depends upon the just packages, like it is a phone and app. You can whatever the functions you need regarding that, you can download the apps and packages and then install it and then use. Okay. So for this library heat map construction, so what we'll do, there are some libraries like library I have written here. You can also install it now. So from here you can install like this. Here you can go, suppose if I type HMISC. Okay. So I have already HMISC. Okay, successfully installed, right? So if you want to check whether that program is there, that package is there in or not in your software, so you can check here, right click and then check it. And this is the, you have to click if you have to use any software or any package in this, actually, and you have to click on just here like this. You can see on the screen. So that means it will be functional, okay, at that particular phase of point of time. So let us see. Library is library. I just wrote library means package library. Okay, just I wrote in library what is required. So complex heat map, circleize. If you just type here circleize, okay. Circleize, like in heat map and correlation analysis, you need so many packages, reshape and so many things will be there. Okay, so you'll require these kind of packages. You can like, okay. So without wasting time, I have to go I have to cover so many things. So let us go directly on the program, and then this is called script. Okay, this phase here you can run your script. You can type your script and then run it. And when it is running, this is console. This phase is called console, and how it is functioning, what is going on, it will tell you. Okay, and if some problem also pops up, it will show. And what are the files in your folder, it will show here. Okay, if any readable files is there which is accessible for this R program, it will be showing here. And these are the here what is output will be coming out, you can see in this console, in this section of this. Okay, so the very first command is let us file choose is there. Okay, so in file choose, so I will not tell you about this script writing, because it will take a lot of time and you need a lot of a lot of time it will recall. You need to practice. Once you start practicing, that's why I have given the whole script. You can go at your home and practice, and if something is there, you have some 5 to 6 days here, you can interact with me. Okay, later on also, if something is there, you just let me know, I can help you out. So if you just click on file choose, and this is run command is there, run is there. Okay, if you click on it, so it will open a window like, okay, run. Okay, so file choose, there should be, see, one window opens directly. Okay, all of you are doing it. Okay, so I need to go to that folder directly. I need to go that folder. Okay, so R programming, I'm taking this file. Okay, so I'm taking this one 30 nph versus 500 nph. Okay, because it has all the names. Okay, full form names are there. Yeah, so you can go to R program here, right? So file.choose I have given. File.choose means it will take you wherever in the computer system, and you can select, go to your file folder and then select from there directly. Okay, so I just clicked on it, my cursor is somewhere here, right? And then I am keeping here run. Okay, once I did run, then it opened a window called this. Okay, it opened in my computer system automatically. Did it come? For how many of you it came? Just click on that. I hope you installed dplyr, hmisc, dplyr, dplyr. Yes, you have to install it. Do it fast. It's easy, na? No. Yeah, it's coming, na? Where you have kept that folder? Yeah, you have to minimize the screen sometimes. That pop-up window doesn't show up automatically. Just sometimes you have to minimize the other windows which is open. Yeah, and then omics data analysis. Open it. Yeah, open. Double click. Yes. Now you have to. You didn't unzip it. You have to first unzip the. I have given you in zipped format, right? You have to unzip it. Okay, I understood. See, I have shared you this omics data analysis, right? If you unzip it, extract, if you right click on it and then extract here, okay? Extract here, then it will get extracted. So since my file is already there, okay, so what is it saying? Yes to, like it will be, you have to rewrite it. Okay, so I am saying yes to all, it will rewrite. Okay, so R program, suppose 3 folders came out of it, right? So you go to R programming, then you opened R programming folder what I gave. Okay, like this. Everyone has, please raise your hand. Everyone is still here. Okay, then go to R program, R software, R studio software, and then file choose is there, right? Very first file choose is there. Before that, where the comma is finishing, just keep your cursor there and then run it. You run it and then just minimize the folder. Okay, if you minimize, you will get this kind of This is pop-up window which has come out. Then through this you have to navigate to your folder. Folder means R program folder. Able to? How many of you got, please? Okay. Or if problem, maybe on one computer, two people can sit so that you can help each other if one is not understanding. Okay, so I am taking it, otherwise it will take a lot of time. I opened, okay. So I opened and then it came as a, here in the console you can see there is a file, it has come as path, right? What I was mentioning, the path, right? So you have to take till programming only, okay? Till programming. You can see in my here in on the screen, you don't have to take all the path, you have to take till screening. Here in the screen, if you see, you have to take till there, okay, and then just copy it, Control C, till programming, okay. And then if you see here, path is another script is the path, right? Path, and so this path I am going to change, okay, because I am changing the path here, I have to change, okay. So I am copy pasting here, so this and this should be under this double inverted comma, okay, fine. So if I go at the end of it and keep here and then run it, so once you get like this in the down console, down section, if you see something like this, you can say that you may be sure that yeah, it is running, okay. If there is any error, it will not come like this, okay. That means it has reached to that window, that folder directly now. After pasting, you have to again run like previous I ran. You can you have to keep inverted comma, under the inverted comma, double inverted comma, and then keep the cursor somewhere like this and then run it, right? Got it? Did you reach to your folder? All of you? Okay. Yes, please. Okay, then let us run the second command. It will take lot of time if I do like this. Okay. Understood? No? See how I am doing. I went here, choose file. I just kept the cursor here, then run it. And after running, it gave me a window. I mean, okay, I should show you again. Okay, it now window. I ran it. Window has come, right? Automatically, this window has come. So from here, I am taking this. I just clicked on it, and then open it. So if I opened it, so now see here again it is showing a path, right? Down. At very down it is showing a path, right? So this path I am copying, right? This path I am copying till here only, programming. Fine. Okay, the whole path you copy. Fine. Control C, I just copied. Control C. If you click after selecting that, you press Control C, and then you go here. The path. Another second script is the path, right? So if you go here on path, select all of it, and then just back, and I pasted it. Control V. Okay. Fine. And here, from here, I am just deleting this much. Just file name I am deleting. Okay. File name I do not want. I want to reach in that folder only, so I have to delete that file name. Okay, and then see, I'm now here, my cursor is here, right, and I am run it. So again it came. Fine. So this is another script of data. It will read my data. Okay, whatever the data is there in my folder, now it has access to that folder. It will access all the files available in that folder, and if I have to save anything, it will directly go in that folder. It will not go here and there. Okay, this is the first programming, means it will not, files will not go here and there, otherwise you have to search again and again and then find it. So it's better to select a path where all the files which will be generated will be going there. Okay, this is the second where I want to read that file, .csv file. So what I want to do, .csv file, there are many .csv file. Okay, so that .csv file, other .csv file you can delete. Okay, so that the computer system doesn't get confused. Otherwise you can directly specifically choose that file also. How can you do that? So I have you can click here. This is another script here. Just click on it, run. Okay, permission denied, it says. Okay, just go again. Okay, no problem. File.choose I am doing again. It is there, and then they are not able to follow. Okay, if I go once by myself and then you will see it. Otherwise, should I first, let us finish first metabo analyst, that is easier. Okay, fine. Let us go online. All of you open browser. Open browser, type metabo analyst like this. Metabo analyst. Okay, all of you got? 6.0, fine. Then click on it and open it. Okay, metabo analyst software is there. Okay, I have given you another folder named as metabo analyst data, na? Metabo analyst data, right? So go to your browser and click here to start, right? Click here to start. Fine. Everyone reached till here? Okay, so there is, see, metabo analyst itself, it is this is web server is completely dedicated to this metabo analysis, metabolite analysis, and you can see a lot of analysis by functional analysis, functional meta analysis, peak annotation also you can do. Spectra processing, spectra you need to process and identify, right? Suppose whatever the spectra you get, that you have to hit against a library, right? Then in order to identify a molecule, right? That is how GCMS or LCMS works. You have to hit it against a library. Okay, so here I will not go for all of it, only 2 of this I will go, and that will be more than sufficient for to get a good data for a sufficient amount of data for a paper. Okay, so first we will go for a statistical analysis, one factor. Okay, this is, you can see here the very first one, generic format, in that statistical analysis, one factor. Okay, so here you can see what are the things, spectral beans you can directly keep. Sometimes this software is also giving you don't need to extract the concentrations. Sometimes you can directly go from the software data which you get from the GCMS or LCMS analysis. Okay, So I'm taking his concentrations now as of now and taking the sample rows in unpaired. Okay, because it has asked for a specific type of data, it asks, and it has to be in .csv format. Okay, .c comma delimited delimited. Okay. So what are the files here? I have all the CSV files. So A versus C you want to take, otherwise A versus NPH. Okay, let us take this file. Okay, because it has the full names and it will be easy for the web server to identify these molecules on the basis of their name and where which pathway they are involved in. Okay. So what I will do, let us go this and take samples in rows unpaired. Okay, and then choose the file. We choose the file. So metabolite data, I will go to GIAN workshop, metabo in a list like this. You can go, right? I'm in the same path, same folder where I gave. Everyone able to follow? Huh? Now I'm taking this A NPH versus D HPH. Okay, open it and then submit. So it will take some time to submit. So data has failed. Why? At least 2 groups should be there. Okay. So there is a very specific format in that. You can edit groups, otherwise what you have to do, you have to take it again. Okay. Here you go to the file, choose concentrations, A versus C, open it, submit. Okay, so what was the difference between those two files I want to show you? Oh, what was the problem in this? Okay, this is the file which I first opened and another file is just now I opened. So that file has been taken. Okay, this file has been taken. Why? What is the difference? This is the file which has been accepted now, just now, na? Okay, there is some sample difference, name in the sample difference. Okay, and when you see the other file, this one, first one which we had taken, there is a labeling difference. Okay, so these small minor errors also. Yeah, that's why. So sample ID is different. Sample ID and sample is different. Actually, sample ID it will take, but whereas sample name is different. Sample here, sample ID and sample, it should be like this. Okay. So here also it has the same name. So let us proceed further. So now it is saying that 2 groups were detected. Okay, in the sample here, this line 2 groups were detected. I will not go through all of it. You can go whenever you are free, you can just practice it. Okay, edit groups. I want to show what are the groups. So here you can see the group label. NPH 33 replicates I have taken, and NPH 500. This is another sample name which is treated. Okay, this is treated. These 3 are treated, and these 3 are upper 3 are the control one. Okay, so just wanted to show. And otherwise, if you want to update or change some name, you can change the name also here and then update it. Okay. Yeah, you can do that. You can do that. 30.1.1 is should not be there. 0.1 was there na, here in this, so that you can remove it and it will get accepted. This one, है ना? Okay, then we'll proceed further without wasting the time. Okay, edit groups, and yeah, I'm hiding it. Okay, let us proceed now. And then so it says normalization overview. So any data, if you have to proceed first, that you have to normalize it. Okay, so here is sample normalization. Here I'm not going to normalize it by sample specific normalization. I will go for auto scaling, which is the best one. Auto scaling, if you read the its a description, this is mean centered and divided by the standard deviation of each value. Okay, so it is one of the best one to be taken forward. Okay. So, so metabolite analysis is not that easy. Also, sometimes you need to do a lot. You need to know a lot of statistics also. What is our Pearson coefficient and all those things also you should know. What is the importance of Spearman rank correlation, and there are so many statistical correlations and theories are there, right? So in that way you have to know those also, but for as of now don't get afraid of that. We'll go for this auto scaling. Okay, it will do automatically, and then go for normalizing it. Okay, so it is normalized, so just we can see how it looks like after normalization and unnormalized data. So this is showing before normalization, somehow its distribution is somewhat like this, whereas after normalization the distribution of the data is bit better. Okay, this is just see, and if you want to for your sake, sometimes they ask also, you can submit in supplementary data also. This thing you can save this from here by clicking on this this icon here, and you can just PNG format or TIFF format, PDF format, SVG, whatever you want to you like, and DPI also, like that is the picture format, like picture resolution. You can select any of it and then submit it, and it will get downloaded. Okay, from here you can download the image. Okay, and you can see and open and see, it will be the same picture. Okay, anyways, so this is how the data looks like, and let us proceed further. And so this is the window where you can do so many things. So here it is telling you the fold change analysis in the metabolites. Okay, so as such fold change analysis you can do by your own self also. It is not that tough. Just treated divided by the control will give you the fold change, right? Isn't it? Right. Okay, so you can do t-test and t-test also, and then for now, as of now, I am proceeding for volcano plot. Okay, so this is volcano plot, and in this there are some statistical things or some modules you can change. So here, like if you see, the x-axis is showing the fold change. If you want to increase much more or you want to decrease, as of now it is 2. Okay, as of now it is 2, and then you can change here 30 control versus treated, or treated versus control, any of the format you can keep. Okay, vice versa. So I'm keeping this one now, and Pearson p value threshold, what you want to keep. Okay, let us keep 0.05, which is more statistically significant one will be selected only. Okay, not all of it. And this is one of the cutoff, which is for biology experiment or like this kind of experiment, it is accepted. Okay, and then let all other be same, and then submit it. Okay, now it is done, and it will show significantly down regulated. As of now, up regulated only these are there, these 3, and if you click on it, it will show you the name. This is acryloglycine. Okay, like let us see, myoinositol. Okay, so this data also you can just save. This is very easy, and there is, if you need it you can save it, but as of now it is not required. You can just save this volcano plot if you want. Okay, but as of now metabo analyst is used for some more other purposes, not for volcano plot analysis. Okay, volcano plot you can do by yourself also in some other different software where you will get a good data, good format of it. Okay, then ANOVA you cannot do, because ANOVA requires a lot of more number of data. These points are the metabolites. You can see here the significantly down regulated, you can see on the screen, and significant. So here it is coming significantly down regulated. Only these 3 are significantly down regulated when you are giving. This is the sample, it is showing. See, it is showing myoinositol is there, and then 2,3-butanediol is there, and this methyl. No, it is not a PCA plot. Volcano plot is showing the expression value. Volcano, like volcano, it's a volcano. Here it is upregulated and this side down regulated. That's all. That is what, na? This is just showing the significantly down regulated. This right side it is not showing. You see the zero value is here. Okay, positively. No, this software doesn't give that one, that format. I mean, completely. It is giving you a kind of just shape of it. I mean, this is not very good for going for publication. That is what I'm saying. Just for their understanding, I'm telling. Okay, but that is what I'm telling. This you don't have to proceed for volcano plot because here it is not looking good. Okay, it is there. Volcano pattern is there. See, from here it is originating from zero. One is going left side, another side is going right side. Here some other metabolites you can find. Okay, and suppose, okay, let us decrease it. Let us keep it. Find one. What happens? Okay, you found some significantly upregulated also when you the cutoff you have changed. Okay, p value. So p value is the one which represents the significantly statistically significant data you can be taken. Okay, on the basis of the p value. Any response is there? Hmm. You are getting where? Okay, double click. No, it is for this single only. N12 octadecanoic acid is a fatty acid. It is telling for this only. So initially we have normalized the data, right, after entering it. So once original concentration they are showing, and after normalization how it is looking like. Okay, we have normalized the data, right? No, where I am not getting. Please, this is for a single metabolite. For the whole set, in reference to whole set, it is showing this particular metabolite. Okay. These points are different sample. Obviously, that is what volcano plot is showing for all the whole set of data. And here you, after putting that cutoff, you are getting this one. These two are significantly upregulated, and these three are significantly downregulated. Others are neutral, are somewhere not statistically significant, or I mean downregulated or upregulated. That's all. Okay. This is to I have to proceed a lot, but just okay, understood, right? Okay, fine. So then we'll go for the correlation. ANOVA is for the more samples, right? You know, right? ANOVA, analysis of variance between more number of data set, not more than at least more than 3. A lot of data is required. Okay. Whereas t-test, you can do t-test. T-test is for the 2. Okay. And then let us go for the correlation. So correlation, if you tell correlation heat map, that is what we were trying to make through R programming. It will give the same here, but here the visualization of data would not be that good, so you can make it nicely in R programming. Okay, from there you can get a good resolution also, and you can do any kind of editing in that. Okay, so choose a dimension, you can take sample, Pearson correlation coefficient. So I was telling you about here, there are so many statistical constants are there, right? Spearman rank correlation, all these things are there. So as of now, Pearson correlation I'm taking for linear correlation coefficient. Okay, and then submit it. Okay. So it comes somewhat like this. So what is this? What is it telling? So here you can see the correlation coefficient bar is there, right? Here -1 to +1. Okay, so here since the value is between 0.5 to between -0.5 to 1, so it is showing like this, and you can see some positively correlating and positively negatively correlating data. So positively correlating as such you are not finding, but here negatively correlating data is there. This whole square is negatively correlating. Okay, so you can see the metabolite 3.1. Okay. Okay. Okay, we'll come back to this after heat map. Let us come to heat map and principal component analysis. And there is one more thing called pattern hunter. Pattern hunter will tell you like there were so many metabolites which are visible here. Okay, you can select any of it. Suppose let us take lactic acid. Okay, so if I submit it and let us see how it comes. So it is pattern hunter. So it is telling in reference to lactic acid, these are positively and negatively correlating with lactic acid. Okay, in relation to lactic acid, their pattern is positive. These ones, their pattern is positive, whereas their correlation R pattern is negative. Okay, so metabolite analysis, you have to have the knowledge of each and every metabolite, which pathway they are coming from. You have to go through the literature, and then you can correlate like from where this which pathway, where there is what is their contribution in the physiology of that organism, that you have to read out. Okay, then let us go for principal component analysis. Principal component analysis is for what? Anyone can tell? Anyone is willing to tell? No. It tells about the variation among the samples. Suppose we have taken 3 replicates, right? So we want to know how much variation is there between the 3 replicates, all the 3 replicates. Because I have tried to do what I'm saying, that no sir, no madam, I have taken all the samples at the same time. Okay, and all of them are good. Okay, and there is no variation. So principal component analysis will show automatically what is the variation between the samples. Okay. So, and that is why it is asked. PCA, this you can use for PCA analysis, and for heat map analysis you can use. It will come. So yeah, this is for heat map PCA. I have used this one, and there are many other formats also. It will show. I will show. So here you can see these are the 2 groups. You can see my NPH data is 500 is more correlating with each other. It is more similar, whereas there is a lot of variation in the first sample here in this case. Okay. Here you can change. There are many views they are giving actually. Overview, then screen plot, 2D score plot. So this is called 2D score plot, and then loading plot. Let us see how it looks like. This is loading plot. This is not that much interacting. Okay, I mean this is not so expressive. Synchronized 3D plots. It's yeah. Wait a minute. Okay, this one is more good. Synchronized 3D plots. It will in 3 dimension. It is showing where are your samples actually, and you can rotate it also. See, the 2 samples are correlating very nicely. These 2 red ones and these 2 green ones. But here there is these 2 one samples are replicates are not very near to it. That means there is some variation in the sample. Okay, And principal component has many components, and the PC1, PC2, PC3 components are there, and PC1 shows the maximum variation. Actually, it takes the max, where is it? Yeah, it shows the maximum variation. Then if it tries to take all the components possible which it can take to assess the variation between the samples, and if something else is left, so other PC2 component PC2 known as the PC2, it will consider the all other leftover components to assess the variation between the samples. Got it? So you can also save the data the same way. Okay. The PC1 always remains the highest one, telling the highest variation, because it is the first component and it tries to take all the other possible features to assess the variation between the samples. Okay, let us move forward. So the PLS, all of these are the different variation of the component analysis, and then you can go for the dendrogram also. Dendrogram, how the samples, these three samples are correlating with each other, how far they are and how near they are. Okay, you are able to understand the concept at least, right? Okay. Okay, then we'll go for the heat map. Okay, it is very easy. You can use it. Okay, and phylogram, where is it? Phylogram. If it is here, I will tell. Phylogram, what it is? Tell. I don't know where it is. You tell what is phylogram. Phylogram, I think it is related to phylogeny, right? Phylogenetic analysis, and phylogenetically how related they are with each other, no? On the basis of genetic variation. So here dendrogram is not taking the genetic variation input. It is taking, I think so, these are the metabolite samples, right? So what do you think, what kind of data set it would be taking to find out the relation? Not the structural. It is telling, dendrogram is telling between the replicates. It might be taking the statistically significant how near they are, how near the values of the samples are there. Okay, so here it is not taking the genetic diversity or the number of the genes or similar nucleotides, but it is taking the metabolite concentrations, how near they are. Suppose one metabolite has a data points of 1 and 1.2 and then again 0.9. Okay, and another data set is having 11, 10 and then 13. So here in these three, the other set is having the more far, I mean they are far. So here the dendrogram will not be very near. Okay, closeness will not be shown. It is just another form of the variation. Okay, so let us submit the heat map. So this is the correlation heat map here. No, it is not a correlation heat map, it is just a heat map. Okay, so heat map, you all know what is upregulated or downregulated. You can see the by the scale here at the right side, just a very upper one, and then for identification which is from which sample data set, you can take this blue and green is there, magenta and red, and this green one, blue is there for the scale. Okay, any of you want to tell something about heat map here? Suppose let us this is upregulated or downregulated. Okay, and this is from which data set? NPH 30 or NPH 500? Huh? Treated. That means NPH 30 or NPH 500? Yes. So you should say NPH 500. Okay, because it is here. So this is how you can identify. Okay. This is the very basic of assessing the heat map. Okay, so these many things you can do in this, and then for correlation I would use some good data set later on, maybe after finishing all of it, because here correlation is not coming that good. So that good in the sense the data set is very less. So I can show you, I have many other files also, I can show you separately, but here I would, in front of you, I would like to load and then see. Here also these are the negatively regulating correlating data, you can see and this is between the samples, between the data sets. Okay, 3.1, 3.2, 3.3. okay, this is how they are correlating between the data set, not between the metabolites. This is features. On the basis of features means you are seeing here the data set. I gave the sample here. Let us come to Excel file. I gave here now 1.1, 1.2, 3.1, 3.2, 3.3. Okay, sample ID. Okay, let us change the feature here. Choose a dimension. I'm going to take sample. There was Euclidean distance. Heat map. Okay, this one. Euclidean is there. Pearson also you can use. It's up to you, like what kind of. No. That is what I'm saying. For metabolite analysis, you have to be bit expert, because reviewer will be asking why you have used this, why you have used Pearson correlation. You should be knowing its significance, its theory. Okay, they will be specifically specifying now why you have used only 3 for metabolites. At least you should use 5 data sets, 5 replicates at least, because there is a lot of variation usually find metabolites in case of metabolites. Proteins and proteins is fine, but metabolites are very dynamic species of living organism, right? Okay, I think I should fasten a bit. So let us go for enrichment analysis. Okay, pathway enrichment analysis. So I'll go for here enrichment analysis, and in that quantitative enrichment analysis. So it will say like these metabolites, where are they taking part, like from where are they coming. Okay, let us go for it. And so I'm going on the quantitative enrichment analysis. Compound names, I'm taking compound names, and then metabolites, what are these metabolites, samples are in rows, and then select it. Okay, so we are here, where are we? In GIAN workshop, metabo analyst. No, for pottery metabolite, this statistical analysis you can do, the one factor what we did, that you can use for any purpose, whether RNA, DNA or protein, because that is statistical one, right? But here this pathway analysis you cannot use. No, only statistical point of view you can have, even RNA, protein, whatever it is. But here pathway analysis, it is going to fetch the data with the pathway and it is taking the metabolites. Let me see once, because I didn't use for protein, just here in case it takes some other, no, lipids also it is taking, okay. Lipids also it is taking. So I don't know if they insert protein also here. So as of now I used for metabolites. So lipid also comes under the metabolites, but the for pathway enrichment analysis for proteins it is not recommended here. Okay, following. Okay, so if there is any mistake, it tells already, it will not let you forward. Huh? So again, if you want to edit groups, you can edit, otherwise proceed further. I have taken 300 versus 500 NPH. Okay, so here what is the phase here coming out? You see, I have taken the full form of the metabolites so that it will be easier for the system to identify and where it belongs. So here it is giving the PubChem ID and KEG ID, and if you see on the details it will tell which pathway it is, PubChem ID and HMDA. So you can know all this. It is easiness for software also to identify if you give the full name of it, and then further proceed further. Then you can do auto scaling here, no need to do all other things for auto normalization, and then you can proceed further after normalization. Which one? Okay, you can read it. So these were not identified, okay, like their IDs were not already available. Okay, so we are going through KIC database, and chemical structure we are not taking, we are taking metabolite name. So we'll take the KIC database, you can take, and for other things you can take other classes of libraries you can use. Okay, and then 2 entries are there, at least 2 entries for a single metabolite is required, and then submit it. So here comes out the pathway. So this is telling these metabolites which pathway they are involved in. Okay, so which metabolic pathway they are involved in. So these pathways are enriched, and here it has taken top 25 metabolites. Okay, for this analysis top 25 metabolites it has taken, and you can see the metabolites are taking part in ascorbate and aldehyde metabolism, inositol phosphate metabolism. So like this you can say, and then in metabolite analysis, as I told, like each metabolite you should be knowing its pathway, where they are coming from. You should be well versed with metabolic pathways and its component species, like what TCA cycle is there or anything is there, whatever pathway. Okay, all metabolic pathways, nitrogen metabolism, carbohydrate metabolism, all other things. So from there you should be knowing the importance of it, and then you can correlate with the physiology of your organism or the treatment you have given it. Fine. And you can save the data the same way, the picture you can save here, submit, and then download. It will come like this, and you can save it, and you can go for the publishing. Okay, this is all about metabolite analysis, and these 4 to 5 figures or 6 figures you will get from here. At least one major figure you will get from here for your. This is one of the best figure you can use for your paper. And there are many other formats of it. You can see they are giving in different formats here. You can like on the basis of the heat, like heat bar. No, you don't have to select. It is telling the overall picture of the metabolic pathways. You should select this red one, which is highly upregulated. Okay, the bar you can see here, na here. Wait, I open. No, it should be. Wait. Here, where they have given. Okay, I downloaded. I think so. This one. Here, p value they have given, na. So this is a negative. This is positive one. This is highly upregulated. This is downregulated. So these pathways are okay. These pathways are highly upregulated. This is downregulated. Yeah. Single one, I think so. You can try, but single one, maybe yeah. Single one you can try, but for statistical analysis, I don't think so because it is using p value. Outliers you should not use. You have to filter it. No, outliers in where? In terms of the concentration or where? I'm not getting. Outlier metabolite, how it can be an outlier? Hmm. No. So when you are taking, you have to take, in metabolite analysis, you have to take the comparative account. If some metabolite is not present in one set, another set it is present, you should not take. You have to neglect both of it. You have to omit it. It will not do. Otherwise, it may tell some error. Because you have to give the similar platform for comparing. Okay, so this has a good resolution, you can use it. Okay, and so should we go for cytoscape? So all of you, hope you have installed it, cytoscape software. Huh? Sorry? अच्छा, टी ब्रेक कर सकते हैं. Yes, we can go for tea now. Huh? Then why did he say I didn't? Ask him then. Narendra is there. Okay, if it is not, then we will proceed, na? I'm opening cytoscape software now. Tea time. Okay, we'll go. Tea is ready. So we can have some refreshment and then come back. Let us get hydrated and then we have come back. Okay. They were like yeah. No. Before setting, there is one control divided by treated or treated divided by control. Two samples here. If you are giving treated divided by control, that means it is highly upregulated in treated. No. In treated.
Treated divided by control. Then treated.
Treated. And if it is control divided by treated, you can change that there in the settings. Before submitting, I submitted. There is one submit button.
Now this is like we have given control and treated. But if I'm doing some phytochemistry analysis of some medicinal plant or something, then they won't be control and treated.
Then how would you say that it is upregulated or downregulated? There should be some control, na? Some base should be there. Pavitra, after this you want to go for thylakoid isolation? No? Okay. Okay. I think tomorrow all of you can practice for this metabolite analysis at your home, no? Tomorrow is holiday, no? Acha, tomorrow. Okay, okay. Calling, no? Okay, so welcome to all of you for this remaining session. Here this time we will go for R programming. We have to finish because I want to show you at least how to make the matrix. Okay, so matrix is the important thing for proceeding any kind of interaction analysis. Okay, so matrix will be based on the p value and r value. P value is the one which tells about the linear correlation between the metabolites or the samples and how significantly they are correlated in a particular direction with each other. So whereas p value tells about the significance, okay, whereas the r value, it tells about the linear relationship between the samples, okay, and how correlated they are, like a correlation coefficient it is, okay. So usually the r value is taken for moving forward and to look for the expression type of things, like so r value is considered, whereas the p value is for making the filtering out the coefficient significant or non-significant one, okay. So so on this basis, first we will make the matrix and we will try to take out the p value and r value, and then on the basis of p value we will filter out the significant one, and then we will proceed for the cytoscape interaction analysis, okay. So I'm waiting for my laptop battery is getting down, just waiting for some time. Anyways, in between what I did, heat map analysis, I did correlation heat map analysis in Metabo Analyst software. So here last you can see the same thing we can make in R programming also, but there we can do according to our own self also. The other one. That is mine. Okay, okay. Okay, okay. Fine. Okay, so I gave you another file of script, R script, which is the making the data for interaction analysis. So interaction analysis we can do from the cytoscape software. Okay, somehow like this. So let us open and the R software and open that that script file. So this is somewhat like this. You can make how the metabolites are interacting with each other, and this is just a representation. I just saw it and it is on present and just showing it. Okay, so somewhat like this you can make, many formats are there you can make like this, and this is just one of it. It was just showing. So let us go to R first, and I hope all of you have opened it. All of you are here, right? Okay, let us set a working directory first. Working directory, how can you set? So file choose, right here, and then run. So in the down section, lower section here, you can see the path, right? So from under the inverted comma, double inverted comma, you can take this and then copy. Okay, copy this, and here now look at the in here. This is the set working directory. Okay, you can, okay, file choose, where did it go? It didn't open the window, any directory, it should open, right? Okay, so file choose I have done, and then after this directory is coming out. Okay, let us cancel it. Go further first. Actually, there are in R there are many ways you can set a working directory. Sometimes I go in that. Okay, I'll be very specific, and just file dot choose, run it. One directory might have opened. Just I'm minimizing it, so it has opened, and I will go to my file. Here. Okay, so I'm taking this file, this 30 NPH versus 500, and just open it. Okay, so this path has come. Okay, file dot choose this, it has given me a path, so I'm taking it. Now you can just copy it, and here in set working directory is there, right? File to beneath the file choose this set working directory, you select it and then just copy paste here. Okay, and here I don't want to select this this file directly from here, so I'm just going till this file name I'm removing. Just see here once, then after that it will be easier for you. This file name I'm just deleting as of now. Okay, till here only, so it will be giving me access to all the files in the folder. Okay, now if I run, so that means I'm already now in that folder. Okay, and this is another one, to read .csv file. So usually R accepts .csv file. So again I'm going to take specifically read a file .csv file. Again I am running it. I can run from here also. In R script you can just, your cursor should be somewhere in the script, and then run it, it will take automatically. Okay, so again I'm keeping it here and then running. Okay, it again has opened the file, and I'm selecting this file now. Okay, 30 NPH versus 500 NPH I'm taking, and it might have taken now. Okay, yeah, it has taken. Okay, so this command down one is the for reading the file. Okay, but when we see this file, data file, okay, so it is telling table has data frame has 52 variable, 52 number of I mean, what is the length is there, and 6 observations of 52 variables are there. Means variables means 52 metabolites are there. Okay, not 52 metabolites, 52 variables are there. Now let us see what is it. You can open it. So okay, here the same file is able to open. Now you can see in the file here in the screen, here the sample ID and everything is being shown, right? So let us come to our here my script again. So correlation matrix. So again, so what proceeding further, for proceeding further, here we don't need this one, sample IDs and sample name we don't need. So otherwise from previous already you have to delete it in the Excel manually, otherwise you can delete in R also. Okay, these two IDs I have to delete, these two columns. So there is a script for that, you can delete it. So so new file which will be generated, it is now going to be called as data1. Okay, data1, and this lesser than and hyphen is there, right? So it says, means it's a kind of data 1 is equal to. Okay. So if I am deleting, so data was my file was read as data. Okay, here it is written as data in this one, written as the data. If I see this file, see it is giving the same file, right? So I have to delete from my data file, which is named as the data. I have to delete these two initial columns, and so what I am doing, data 1 is equal to data, which is previous one, data minus column I'm minusing from here. I'm reducing here, and then just do it, run this command. So now data 1 has generated now here, and if you want to see, now the two columns will not be there. You can see any problem? Hello, any problem? Please be concentrate. I'm giving my time here, so please be okay. And if any problem is there, please let me know. So data is there, and then this is the correlation matrix script. Okay, this is the script you have to make, like with time you can learn it, and as of now I have made it for you. And so Pearson correlation matrix I am making, where I am taking the data 1 as a input file. Okay, data 1, see I have written data 1 here, data 1, right? You are able to see the file, and this is data 1. So it is going to make the correlation matrix on the basis of data 1, and if I run this program, the correlation matrix will be made. This is the R core it has made. This is the correlation matrix it is made here. Okay, so and from this correlation matrix we can take out our R value and P value. So for R value is equal to correlation matrix I am taking out from this correlation matrix, which I have already made. Okay, so if I run it here just I kept my cursor here and I'm running it. So this R value it has given me. Here you can see it is the R value it is giving me. Okay, so you can see, let us see and open and see. So it is giving the R value. You can see this is the metabolite names and how the R value it is. Okay, so let us come to script again. Then let us go for the P value and run it. Okay, this is the script for the P value, and again I'm running it. So P value also might have come here. This is the P value. Okay, so once R value and P value is there, but we have to save it also, right? So we can save it, write.csv. Write.csv is a command to save. Okay, write.csv, this is the whole script. So how I'm going to write it. My file name is, just a minute. What is the file name is 30 NPH versus 500 NPH I have taken. Okay, fine. So I'm going to take this one name. So this name I'm copying and I'll paste it here. Okay, .csv. Here also .csv. So it will be saved in this name. Okay, so let us run this command, and okay, it is taking what it is saying, component file 30 NPH permission. Just a minute. Permission denied, it is saying. Just a minute. So here R is very, as I was saying, R is very specific to any kind of variations or any mistakes. It will take it directly and it will not run. Okay, so what I will label it, I think this 30 NPH it is not taking. It is saying some for this permission is not available. So I have to change the name of it, and sorry. So 30 and pH, let us consider if the same file is there or not, I need to see. Otherwise A versus C, P value and R value is there. So in order to variate it from previously available data, I'll just say it A versus B. Okay, here I'm just, you can see A versus B, A versus C, A versus B, P value, A versus B, R value. Okay, now let me run. So now it might have saved in my folder. Okay, let me see if it is coming. If the directory was set, it should have come. A versus B, R value, it has come here. Okay, so it has been saved now in the my set directory. So again now write the .csv p value. Again if you run this command, it will be saved. Okay, so let us just check again with p value also came. So R value and P value has come. So now let us see, open and see how it is looking like, R value and P value. Okay. So this is the P value. Okay, P value matrix, and another is R value matrix. Okay, this is how it looks like. No need to think much here. It is just looking like this. I want to show you. Okay. Then further, what we have to do, since how these metabolites are interacting with each other, that means suppose one metabolite is there, it is, I'm sorry, okay, one metabolite is there, it is interacting with 5 members, 5 other metabolites. So this one metabolite has to be written 5 times against all these 5 individuals separately. So I have to make a file in a linear column that suppose one metabolite A is interacting with 5 different metabolites. Suppose column A in Excel sheet is there, suppose column A is there and column B is there. So column A, suppose metabolite glycerol is there. Okay, glycerol is interacting 5 different. So I'll write in column A glycerol 5 times and 5 in column B 5 different metabolites against it. So now I have to reframe it in that way. If I do it for 80 number of metabolites separately and manually by my hand, like by sitting and doing it, will take a lot of time. Okay, so we will write the script for that and do it. So that is the file format which will be used for the cytoscape. So for that, what we are doing, we are reshaping it. So for reshaping, since what we are going to take, we are whatever the R values and P values we have saved, we are going to use these file names. Okay, so these file names again I am going to copy because I changed here. So this is P value. Okay, this is R value. Right. Okay, so A versus B, P value, I am reshaping it. So let us see how it looks like. So I ran it, reshape P. Okay, so you can see now it has read the file. Okay, we have read the file. Reshape P, we are reshaping it, but for reshaping, and the name of reshaping, I am reading the file. Reading the file in the name of, now in the R environment, the file is known as the reshape P. Okay, my file is the P value of P value file. Okay, but in this environment which is existing now in R, there it is known as the reshape P. Okay, so again adding one column of metabolite name under the ID column. Okay, if I run this script, there will be a change and an ID will be generated, reshape P ID. Okay, And again, if you run reshape P here, propanoic acid was the first one. If you see, the first metabolite is the propanoic acid. So I am making the ID before propanoic acid. Okay. So here the command I wrote in that reshape ID, relocating the ID before propanoic acid because it is starting from propanoic acid. Okay. Then run it. Okay, before propanoic acid, I don't have the function reshape P. Okay, so it is saying that I don't have this software reshape P. Okay, so what I'll do, I'll go for here reshape. I have already installed it, but it is not in the current working environment. Reshape I have already installed, reshape 2 and reshape. Now it is in working environment. Okay, so reshape P. I was running this one, right? Reshape ID. Yeah. Okay, again is what it is showing reshape relocate ID before propane could not find the function. Just a minute. No, oh, no. Okay, there is some mistake in the file selection. A versus B. Okay, so it is better. Let us make a new folder just to avoid confusion for R also. So, new folder. Okay. Control C, Control V. We have to run this all this again. Okay, it won't take much time for me. Just file. I'm running file. It won't take much time. So file, and I have to take new. Sorry, where is it? A versus C. Just a minute. Let me see. A versus. Okay, fine. Okay, so I have taken the file. New folder. Till new folder I am copying. Read it. Okay. File.csv. It will again come. I'm taking this. Okay, data is equal to this one. So now the files might have come already in this, right? A versus B and A versus C, everything has come. And let us go for reshaping it. Okay, propanoic dot acid. And what I have written here, propanoic acid. So sometimes R create problems, okay? So it is saying ID dot before propanoic acid, unexpected symbol in reshape R percent relocate ID dot before. Okay. Just a minute. Okay, I'll work out on it. But before that, let me start with cytoscape once. I'll give you, I have already given you the data for cytoscape. In between, I will tell you how to do this. So you can open all your cytoscape once, please. So in cytoscape, you can go to cytoscape data is there. Filtered and raw data both are there, right? So in cytoscape, let us take you can go to this icon here, or else you can directly you can also take the file from here, import file, import from file, okay, and filter data. And then filter data means I have put the p-value significance 0.05, 0.05, okay, p-value. On the basis of p-value I have filtered. And okay, let us take as of now filter data or raw data, any of this you can take. I am taking the raw data, you can take filtered one, okay, it is easy. And so here now it is going to be created. This is what the file I wanted to create again in Cytoscape, sorry, in our program. So you can see the propanoic acid is many a times, okay, and it is propanoic acid is interacting with other metabolites, okay, first of all with itself and then again all other, you can see here on the screen. So it is interacting with all other, okay. Then after some times propanoic acid is gone, then it's the turn of 2,3-butane diol. So 2,3-butane diol is now again interacting with all this set. Likewise, now again comes the lactic acid. So this is what I was trying to create with R. So we'll in between I'll give you this work, and that problem I will work out and we'll do it, okay. So in between now what you have to do here, you have to take there are two sets, two columns are there. One, you have to take the source ID as the source node, and another is as the variable node, okay. You have to come as variable, and then this is the variable will go as the target node, okay. One is source node, another one is the target node, okay. Now we have taken imported it. So this is the network coming out, okay. So this network we have to make it, but its name is not coming. Why? Its name is not coming. So in filters it is coming. Maybe for me the I think labels color is same, otherwise I think so. Label color, let me see, label color. Let me see, label color black. Okay. Okay, for that. Okay, if you have taken the filtered data, fine. Names it would be visible, right? Okay, so you can arrange now it is a very time taking process to make it in a network format, in a very publishable format. You have to pay a lot of time here, and by sitting you can here by the time I'll work on R, and you can, I will tell you how to do this interaction analysis. So you can somehow make arrangements so that it can look good. Otherwise, so now we have imported it. So look at here first. You can go to style. There is something is happening. I don't know. Style. Okay. Network style is. Okay. So style you can change whatever the style you want from here. Okay. You might be getting some more styles, so you can use accordingly whatever style you like. And then all of you, do you have CytoHub? CytoHub you don't have, no? So all of you can go to app and then app store, okay. Show app store, and in app store it is not there, I think so. So you can go app store, install from app file. No. CytoHub, type CytoHub in the, if you have gone to app store and show apps or install apps from file. Huh? In the Cytoscape only, here in the apps you can go, okay. CytoHub. CytoHub like this should be there. cytohub, like this. I can share you this screen otherwise on your WhatsApp, and then install. Otherwise you can directly go here, app manager. Is it coming? Yeah, for you it came now. If someone, since I have already installed it, you can go here, app store, and then show app store, and then type cytohub. So it will direct you here, cytohub, like this. So in this, in my case, it is like this. In your case, you can install here. Yes, yes, you are right. Yes, installed. Anna? Yours is installed. Now fine. It should be visible now. Wait na. Cytohub you installed?
Yes.
That's great. This one, fine. So it should come here in the center.
Already installed.
Here it is, na?
Ah.
Okay, like she has got, how many of you have got? Got all of you? Okay, then you should tell now, that's why. Okay. So here we'll do one more analysis here. Analysis is go to tools and go analyze network. If you do this analyze network and then select OK, so it will tell you here how many nodes, how many internodes are there, how many edges are there. So it will tell about the summary statistics, number of nodes, number of edges. So what are the nodes? These are the nodes. Okay, these are nodes and these are edges. These are called edges lines. Okay. So now from here we can go to, so this is the whole statistics of it. And now you can go to cytohub. Otherwise there are many other things also. Styles you can change here, default style. As of now it is showing this, so I'm proceeding with this one, and I have already selected it. And you can, if you want to change anything here, color you want to change, like you can play around with this. It is like a sketch board. Okay, so you want to change the color, you can change the color. You want to border width, you want to change. Border width is 0 here. Now you want to keep 4. Okay, border width 4, you want to keep 4. So now you see the border has come. And then border color also, if you want to change, you can change it. Border width and fill color, and then this color is specifically if you want to change for any particular one. Suppose for this one you click on it and then fill color here, this one, left side, right side one, this card. This is for overall, this is for individual one, this box. Okay, and then suppose if you give it for fill color, suppose this one you give and select it. Okay, so now it will come for this, specifically for this one. Okay. Likewise, if you want to change the label, you can change the label name also, and then label color if you want to change, you can change the label color. You can just select from here. In my case, I don't know, something has happened. Okay, let us keep this. Let me keep white. It comes. there is some problem has occurred in my laptop. Anyways, so all of you are getting the name right? Okay, so this is all, and label font size you want to change. You can change the label font size and shape. Then shape if you want to change here anything. Usually eclipse is fine. Ellipse. Hexagon you want to apply, you can use hexagon. Okay. Then size and all this. After this you can go to CytoHub. Sorry. Anything. You can go to CytoHub if all of you have then calculated, and then after this you can see how many metrics are there, how many. Okay, modulated in CRC. There is 24 nodes are there. So maybe you can select any top of it. So you can come to CytoHub here, and you can take maybe let us say top 20 or 20 top 10 or 20 or 15, whatever you want to take, you can take. I am taking top 20, and this is kind of usually for the papers, this is kind of degree of interaction, and this is kind of where we use Pearson correlation coefficient or R correlation, right? Or Spearman rank correlation. In that way, these are the bottleneck, eccentricity, closeness. On this basis, any of it we can use. And so usually for the papers, MCC and degree is accepted. Any of it you want to use, you can use. So then filter it on the basis of check and first edges and nodes, and display the shortest path and submit it here. Should change. Did it change for someone? Right? No, it has to change completely. Like it's, yeah, that one is good. For me, I'll do it again. Okay, I don't know, somehow it is not coming. So all of you continue. I'll turn it off and then. Okay. So in CytoHub again, it will give you in a very directed format. Use CytoHub, you can use now. Installed?
Yeah, install.
CytoHub. Turn it on and off again. Close it. Sometimes it happens. Show me.
Top 10.
Warning, please input the name that you are interested in the text area. Okay. No, here you don't have to select degree. You go MCC. No.
Same icon.
What about degree? You can change
color.
You can take this, so it has given a degree.
It's MCC now.
No, not a problem. Something different. Anyone else? Yeah, yours is good, na? Here. Name somehow I was okay. Anyone else? You used filtered one or like filtered data? Okay. Till now I was getting. Anyone else? Anyone else got? Okay, I'll show you my saved pictures. Let me do it again. Sometimes software, I don't know, it is happening. Import from file. Okay.
Previously, filter data was done.
20. Okay, those who have are running in their system, please close it. It takes a lot of space actually while running, so that may also be a problem. So network style. So now I'm getting, yes, there is a problem. R, if R is functioning backwards, please switch it off. So nested network analysis, you can select like this. So this time I selected unfiltered data only, but the name is come. So it was because of the occupancy in the system.
You can move like this.
You can move, you can play around like this. You can. So how in published format it will look like, I will show you. So in that way you can arrange it by here, time being. Downloads. It will look something like this. Something like this it will look like. Okay, you can make like this, and here there is a gradation also will come, I will tell. So those red ones which are highly correlating, highly interacting with each other, these are highly interacting with each other, whereas the gray ones, and also you will get a value like gradient value, I will tell. So on the basis of that gradient you can select the one which are having more number of interactions, that size you can increase, and then in gradation you can decrease the size of these nodes. Okay, and so somewhat like that it will look. So I am going to directly CytoHub and calculate, and then top 10 MCC, display, submit. Okay, yes, for me it came now, and then you can go to you can go to style, nested network style. Okay, so it is just kind of a game or something like that. So you can play around, and then you can change the name also, everything, whatever way you like, and you can refer to so many papers also. Okay. So has anybody selected the filtered one? Okay. Did you find any gradation here in the rank? Gradation in the color at the right side, rank. No one? Yeah, one rank means all of them are equally interacting. Okay, so maybe just let me ask who is getting it. So here there are many things. Layout you want to change, you can go to layout, and layout circular, you want to see the circular one. Okay, and then there are many formats you can check out. Hierarchical one, hierarchical doesn't look good, it is so different. Then you can organic layout, this is organic layout, orthogonal layout. It may sometimes since your laptop it requires to install so many things in between, so it takes out automatically, and then so anyways, and then further, so you can see here this is the border width you want to change. You can change to 4 or 5 like this. Then you can change the label. I want to change the label suppose for what, for which one I want to change, suppose for propanoic acid I want to change the label. I want to say PPA, so you can change like this for individually for this. And further shape and then transparency also you want to change, so you have to make it look in a good look, so that it looks fascinating. At the same time it also gives the information. So suppose if you have to give what information you will fetch out, you had the metabolites, you had got some concentrations, so now those concentrations and metabolites are in this network format in front of you. So how would you analyze now? You now know that they are interacting with each other, right? And they have got these many nodes, these many edges they have, and so how you are going to analyze? Like these many edges are there, edges, and these are lines are called edges. Okay, edges also you want to change, you can change in the same way, like it's a label color and all those things. Line type you want to change, you can change this here from here, anything you want to keep, so it will look like this something. Okay, so I think sometimes solid looks better, this one. Okay, and further let me select the other one. Here I am getting all the correlation or the interaction to be of rank 1, so that is why all of them are labeled as red. So let me, I'm thinking to take the another data set. Let me take the filtered data set if something comes, and this one local, and then I'll go for sorry, this is source node, and then you can go for the variable, this node as a target node. Select it, let it come out, and then just change the style to nested format. You can do the same thing again. You want to change, and then further you can go to CytoHub, so you can change in I have taken this one is going on top 10 MCC or degree, anything you can take, submit. So here also it has come, maybe let me take for top 20. Okay, how many edges are there? 24 are there, na? So almost all of them are correlating, interacting with rank 1. Okay, so otherwise sometimes what happens, there will be gradation, like A few of them, the top ones will be interacting with rank 1 and some with rank 0.8, 0.9. In that way, you will find a gradation here. Let me select another file. Otherwise, in this case, I have sheet top 20. Okay. So while exporting, you can export it through and layout tools also you can change here. There are some layout tools. Change. It is not changing. Not sure. Sorry? I don't know Telugu. So like this you can do, and while saving, let us see how to save it. Suppose I have taken top 10 one, top 20 and submit it. Okay. So you try to do, and for edges also you can change, and there are many more things in this, like let us go to style and then transparency, width and all this you can change. Let us go for edges, edges now, and edges in width you want to change. So let us go for column type, interaction, and then mapping type, discrete map. This one you can go, otherwise you can go for discrete mapping. And sometimes if you want to change the color of a particular Stokes color you want to change here, Stokes color, suppose let us go for this, can change here.
Oh, you highlighted that 4 color.
No, yeah. So all of you able to understand this here? Huh? No, highlighted means here the red one. What are the red ones? Huh? What are the red ones? Please tell me. They are having the highest correlation, if not correlation, interaction. Okay, with rank 1. Okay, all of them. There is no gradation. Otherwise, some other files, if I have, I should select it. So let us see how to save it. You can save it from exporting, otherwise directly from here, export file to image. And usually PostScript files you can save, and but you cannot directly open that. You have to open it first, convert in PDF and then open in Photoshop. You can work out and you can have a highest resolution. but you can directly open in PDF also, and then later open in Photoshop. That will also give you a good one, good picture. So let me save it at folder A. Yeah, okay. Okay, I'm going to save here file name like trial. Huh? Okay, so it should come here. It has come. So like this, it has a good resolution also. You can use it as much you want to zoom it, you can zoom. Okay. So please try to make a good interaction network by using all those in between. Then we will come back to R, and I'll come to you and see how it is. Yeah, that is just for, since you are on there, that's why it is changing, but when you go up, it will show its own color. Okay, so it is just by default, it doesn't matter. Okay. Hmm. Hmm. No, click out of the box. All of them are red, right? So when you will save, so it will come as red one, all of it. Okay. So when you are clicking on something, that system is showing that you are concentrating on it. Now you are working on this. Suppose you want to change its name or something. Let us go for nodes. Here node and edges. If you have to work on nodes, these are the nodes. This is node, and these lines are called edges. Okay. You have to do any modification in nodes. So you have to come here down, nodes, here down, nodes are there, and then you can, suppose, let us change its shape. Okay, I have selected this. I am going to change its shape. I'm going to make it circular. Apply. Okay, so only this has become, since you were, that's why the color change occurs. Now I am going to concentrate on this. So see here, propanoic acid, right? Although its rank is 1, so here, because of rank, it will be there. Since I am going to concentrate it, it is now centered, right? So I can change to something else, or I can change its height, width. Let us change its height, particularly of it. So let us make it 50, 60. Let us make it. This is 60, and then its width I need to change. Let us make that also 65 around.
So
Sorry.
which option we can use for the high correlated?
Okay, and then you can again change. Suppose myoinositol has the rank 1. Okay, rank 1 myoinositol, where it is. Here myoinositol is this one, so I'm going to give it a some different color. Set bypass, I'm going to give it a very blue color. Okay, now it is blue. Okay, and then I'm going to give the highest size. Suppose since its size is too much, like its rank is very high, so myoinositol, its size, height no, height will change here. If you are going for the individual one, so these right side columns are there, and if you are going for all of it, then right side one, left side one is there. Okay, so that you have to play around, and then in that way make a good interaction. And you can also suppose myoinositol is the highest rank holder, so it will be given, you can give a big shape. Then lactose is there, it is also having rank 1, but it is second in this rank. Okay, The top one is the glycolic acid. Okay, so suppose you have a data where you get all of them to be in some gradation. Okay, there will be gradation. Suppose you read less red, and then likewise there will be gradation. Okay, like we found in the heat map analysis. Okay, so in that way we can change, and then you want to, then you can go to edges and then change it, the line type, level, font size, color, everything we change. So try to do it in between. I'll come back.
జగనన్నలాగా అకౌంట్లోకి డబ్బులు వేస్తున్నావా జగనన్నలాగా అకౌంట్లోకి డబ్బులు వేస్తున్నావా which option you have to select to find the high the red color ज़्यादा है ना so how to change that option
कहाँ पे?
इसमें
Change it now. Select it. Click. You selected. Now you change the color. Go to style. Style.
not individually
All of them.
all of them
So all of them right side on. If you change for all of them, so this rank you can use. If you have to change for individual, you can use this. Anything.
okay.
Suppose this
For example,
shape. Can want to keep this.
which metabolites are linked
Uh,
interaction with myoreticol.
Wait. Only this change, shape.
Okay.
Export it now.
Import.
Not have deep format.
ಏನು వేరు ఎక్కడికి ప్రోగ్రామ్ అవుతుంది ఇప్పుడే రెండైనా అంతే ఇప్పుడు ఆ ప్రోగ్రామ్ మధ్యలో స్టక్ అవుతుంది ఓకే Here now it's showing actually. So you have to show, oh here, okay. Now I got it. From this what we will do? This one now. Then no need to do the first is now
1 2 1 2 1 2.
what about this? Why? This one? So okay. So this means what? Form of current rank. Okay. This is what I was. Now I got it. So we have to say this. So can we get the description with this software?
Hmm?
Can you get the description of image?
Description of image is not there. You have to describe by yourself.
We need to add the AI.
Yeah, you can do that.
Okay. చెప్పడానికి ట్రై చేస్తున్నారు. అవునా? ఫస్ట్ అంత ఈజీగా అర్థం కాదమ్మా.
Okay. Okay.
प्रॉब्लम आ गया भैया
नहीं ठीक है. So all of you got it right? Huh? Which one? हाँ. Like which one are you asking? No, this one you have set working directory. You have written something. It doesn't have to go this long. Okay. You just cancel it. Set working directory space less than equal to this one. Now it will go. Fine.
These are, how do I debug this? Should I check for column and space and all that?
Yes. It's running now. It's running. Then see here.
I'm not getting any file.
No. See here. You have here.
Unexpected.
Just a minute. Okay. You got it. So now in that you have to choose file. Okay. Now command might have opened. Wait, just I'll show you the heat map once and then I'll come. Just a minute. जो तो टाइम हो गया, just give me some time. इनको एक okay. Sometimes it occurs.
No need to change your name. Just you have to change your name.
Yes, okay. Okay.
No. One is doing like that.
Okay. Yes, you should not be doing any mistake in R. Okay. So here let us see. File dot choose event. Okay. And then I'm going to make you show you how to make the heat map analysis, cluster analysis in this heat map cluster analysis. Okay, hierarchical cluster heat map analysis. Okay, so file choose what mistakes you if you do here na see by mistake I was not using this one, just this. So if you delete anything, it will show this. Okay, what she was getting. So it should be always under the comma and under this. Okay. So if in hurry bhari or something like that you work, and so it will go off. Okay. So here you have selected it. I took the file, run it, and then this is numeric matrix. Numeric matrix. For dendrogram, it will make hierarchical cluster analysis. Means hierarchy also it has to make. It has to make a dendrogram. So for that, numeric matrix has to be created. So you can select all of it and run numeric matrix. Okay. So it has run, and then since in heat map you need to give some different colors also. So for that color function, you can select this and then run it. Okay, so color function says this color ramp 2 is not there, so you can do COLOR color ramp 2. This is another library for preparing the heat map. Okay, so once I have selected now, so this is color ramp is taken, and now if I run, it should come if any mistake is not there. So it has taken. Okay, and then creating dendrograms. Okay, since first I made a numeric matrix which is the matrix p value and r value basis only, and then after that color function you have to give. Color function you can change also sometimes, green, white or red. Suppose if I keep it here, you want to change something here, some color you want to keep, so let me keep it blue. BLU. Okay, so if I run it, it should come. Okay, and then create a dendrogram for it, and then heat map. For heat map, you can select, this is the heat map command, it is quite big. So name Pearson correlation I am using. Col function is another col function, like you have to go through it, like slowly you will go, you will understand it. And for the things which you can understand is here, like font size, like row names, gap, and then font size. Okay, so font size you want to change, you can change font size later on. First let us make it and then we'll see. Okay, and you want to give it some title, you can give them title also. So let us go for the heat map analysis here and then run it. Okay, so it is saying here niche down it, there is some problem in running here. So it is saying col function cluster row is equal to row dependent, heat map is not found. Okay, so what I have to do is heat map, I have to go heat map or fit, and I have to, so it is getting installed. So once red comes in here, it means it is getting installed. So now if I run, it should come. So it has come. Okay, so heat map has come. If you go to plots, so it is the heat map. So this is how it looks like. Okay, this is the correlation analysis. So now if you see the advantage of this R, suppose you run all of this in a single go. Okay, I delete it. Yes, okay, I deleted this, and then file dot choose. First I have to do file dot choose, hide it, and then desktop file. You have to take R value for this, any R value we made, right? R value files were there, we saved R value. A versus B, R value I have taken, and then so this is the directory. Okay, this is the directory, I am copying it, and then Control C, and then here under this inverted comma it should be there. If you do any mistake, it will not take. Okay, even a small space or bracket is not there, it is not going to work. Okay, now it is working. And then read the file data, read the file dot csv, so it will take run this also. And then again if I run it in a single go from here to there, let us see what happens. It's done. Okay, heat map came. Fine. So like this you have to do, you try to practice this, that one also, there won't be any such problem. And you can export this file as PDF, agar save as image also you can do. And let us change the font size otherwise. Okay, let us save once first. Path, let me select its path. Okay, sometimes my, okay, let us keep it desktop file. Okay, save. So and let us export PDF as well. PDF directory I have said the same one only, and then save it as rplot1. File name is rplot1. Let us see in desktop file. Just sort by date. So this one PDF. This is how. So how to analyze this? This is the main thing. So you can see the metabolites are interacting against each other, and this, so there are some different color of squares are there, right? P value, what is the P value threshold? P value R value threshold. R value threshold is from -1 to +1. Okay, and in that, suppose there are some squares you can see very these four are correlating, highly correlating with each other, okay, these four. And then if you go this whole big square, these are positively correlating with each other. Positively, why I am saying positively? Why I am saying positively means it is red in color, okay. And then so you can look around for these metabolites, what is benzoic acid, 3-hydroxybutyric acid, silanol, and butane dioic acid, okay. So these are interacting with whom? With, they are interacting with this silanol, benzoic acid, okay. So in that way there is a highly positive correlation data is there, then there is a bigger square is there. Then if you go on top, here is another big square, but in that you can select again this one, and then otherwise you want to take this one. It depends upon you. You will have to have the knowledge of all these metabolites and their functions, okay, like what is their function, where they are interacting. And then again suppose this one is the 1, 2, 3, fourth one is interacting with last. No, let us go for third one, N-methyl. This one N-methyl is third one, is interacting negatively with trisaminol, okay. Able to understand something, okay. So this is interacting negatively, whereas it is interacting like suppose tromethamine is there. Tromethamine is positively interacting with this. What is this? Tromethamine only, okay. But if you have to see, it is also interacting with this very much positively with this one. What is that? This is third one is N-methyl. So it is interacting positively with this also, tromethamine, okay. So in that way it has to be analyzed. So provided you have the you have to have a big knowledge of all these metabolites and their functions and everything. So by making use of R within single minute, within a fraction of minutes, you can just run it, make it, okay. And also you can change its how to say, suppose you want to change its font size, let us keep it 10, okay. And here also let us keep it 10. And you want to change the color, let us make it blue and green, if you want to make. So let us run this again, and as of now I'm deleting it. So if you run it again, it should come, okay. This is all, but for writing the commands it will take lot of time, and you have to put your efforts in that. And see, again the same thing has done, but the color has changed, and you can keep in different formats, okay. So R has a good application, and you can make use of it in so many statistical analysis, and so many things you can do. Just a lot of scope is there, and lot of requirement in companies and other places also. But you can see how sensitive it is, the R script, okay, how sensitive it is. Like sometimes like you get stuck in that and for longer time. So this is all for today, and Jyoti, time is over, right? Or some more time is there? No. And then for today, if any question is there or something you want to ask, please be free to ask.
I'm getting all these.
No, you have to set your mouse.
From here I selected like how we click
File, run file, huh? Where is our file? File dot choose. Command should come now. Is it the command? No, it is not. Command is not coming in your slide. File dot choose.
Thank you for practice.
Yeah.
When I reach home, I'll practice.
Yes.
I should do
It is coming like. So I shared you the data now. Where is that? Where is R programming?
in
So there is R value is there. This one. Open this. Okay. And this file is there. It will come. Wait 2 minutes. Control C. Yeah. Yes, I know that. Okay, and it has to be closed also. Hmm. Please go ahead. Here you haven't closed it. That's why. Okay. Here. Run. Now it's fine, na? And then select this one. Run.
for me.
Okay, do one thing.
I directly went open the file which you gave me now.
D versus in just a minute. No, some mistakes are there in the file. Otherwise, where is that? R program. Where is that? Omics. It's fine, na? Is it .csv file? Yeah, that's why. Properties. Where is show properties? No, it is comma delimited only. CSV. Okay, there is some mistake in that. After that, you run. Huh? I will. So I will share you my file directly today. Which one I used here for heat map? You used that file directly. Okay, for your practice. So cytoscape file you all have, right? Okay, and I'll share you another some other more files reshaped. It will be like reshaped R value, so that that file you can use for cytoscape analysis. Okay, and maybe you can find some gradation in that, maybe. Okay, just 4 to 5 files I will share, and you can try in that cytoscape directly. And for for this heat map analysis, I will share the file which I have used now, and that without doing any mistakes, as a script I have shared, and don't delete any, suppose these brackets are under brackets, all these things. Okay, so it should be very same. So let us, if you want to run any more time, if you want to keep some black here, black here, and then run it. So it will come as black. Okay, here. So so this is all about, and all the best, and see you tomorrow.